Good morning, everyone. And as some people are in the waiting room just joining us, and we'll give it another minute before we start for the day, but we're really excited that you're here for the Rare Kidney Disease Foundation's 4th International ADTKD Summit. Today is the science program, tomorrow's the patient program.
Come back down. When they checked the RNA transcripts.
Okay. Dr. Blyer, preview of what you'll see later this morning. All right, I know we've got people joining. I'm going to get started in the interest of time. Oh, all right. Tony, if you're able, go ahead and mute yourself. And we'll go ahead and get started. Again, welcome everyone to the 4th International ADTKD Summit. ADTKD, of course, stands for Autosomal Dominant Tubulointerstitial Kidney Disease, and that's the name given to how this particular set of diseases presents. My name is Sharon McRoder, and I'm the Executive Director of the Rare Kidney Disease Foundation. Good morning, John. We'll see you in a little bit. Or RKDF is, you'll hear me say RKDF a lot today. I'm also an ADTKD patient. So my grandmother and my mother passed away from undiagnosed end-stage renal failure. My grandmother was 62, my mother was 57. And I have numerous aunts and uncles and countless cousins and 3 of my 5 siblings who also have the MUC1 gene variant of ADTKD. So I also have 2 sons, and chances are 50-50 that each of them, or at least one of them, also have this gene variant. So I joined Rare Kidney Disease Foundation about a year ago and recently became its executive director because I care deeply about this issue, about this condition, this disease, this set of diseases. And I want there to be a cure, or at least a treatment that will stop the progression of the disease so that my sons can lead a full life, as well as all of the rest of us who are diagnosed with ADTKD. So I'm deeply motivated and excited to be here today. So let me just explain. RKDF is a patient-led, patient-focused nonprofit organization dedicated to halting the devastating impacts of ADTKD by supporting physician scientists in their quest to find a treatment and eventually a cure. We are all volunteers, and we all have or have a family member that has ADTKD. Our mission is to raise awareness of ADTKD among both patients and nephrologists, and the entire medical community, actually, to educate patients and nephrologists on options for genetic testing, and third, to help build and connect the ADTKD patient and medical communities. Our ultimate goal is to help Dr. Blyer's Rare Inherited Kidney Disease research team at Wake Forest University find our ADTKD patients for the patient registry, and 2, that registry will then provide a patient pool for a clinical trial of promising treatments currently being investigated by Dr. Anna Greco and her fantastic team at the Broad Institute of MIT and Harvard. This summit would not have been possible without the contributions and undying support of a lot of people. First of all, the hard work of RKDF volunteers, Suzanne Kelly, Sue Reel, Kathy Stankus, our social media and advertising efforts of Lisa Parcell, Christine Upton, Joe Morden, and Joe's fantastic marketing team at the TMV Group. Or the thought leadership provided by our partners, Kathleen Blakesley, who is an RKDF trustee, Tony Blyer, Kendra Kidd, and Stephanie Cloud from Wake Forest University, and Katie Liguri from the Broad Institute of MIT and Harvard. And of course, I have to thank the overall guidance provided by RKDF founder and board chair, Richard Nelson. So thank you all. All right, couple housekeeping before we get started. First of all, attendees, all of you will be muted throughout the summit, muted and your videos off. But don't worry, we do want to hear from you. We'll have a brief 5 or 10-minute Q&A after each session. So to contribute to that Q&A, toggle over at the bottom of your screen where you see the various icons. You'll see a Q&A icon. Please post your questions there anytime throughout the summit, throughout the sessions. And if your question is aimed at a particular panelist, please mention that in the Q&A. And if we don't get to your question during the summit, don't worry. We're going to go through all of them afterwards. We'll connect with the panelists and we'll make sure your questions get answered. And all of those will be posted on our website after the summit. So we have a lot to cover today, and I'm going to be fairly strict about our schedule. So I ask panelists who are already on to grant me some forgiveness if I need to interrupt you to move us along or keep the Q&A moving. Now, if you're having any technical difficulties during the webinar, please email info@rarekidney.org. That's info@rarekidney.org. That's only if you're having technical difficulties, and Suzanne will help you. This event is being recorded, and the recording will be posted on the website at rarekidney.org after the summit. All right, well, let's begin today. Actually, so today and tomorrow, we're going to begin each program by hearing from ADTKD patients about their experience with this disease and their efforts to find a diagnosis. So to kick us off, I would like to introduce ADTKD patient, RKDF volunteer, and registered nurse, Vani, who will share her diagnostic journey and how she was finally able to get genetic testing. And remember, if you have questions for Vani or any of our panelists, please type them in the Q&A box. Thanks for joining us, Vani.
Hi, thank you, Sharon. Thank you for having me, and I appreciate all of you for taking the time today to listen. We really appreciate it. Just to start off, both my parents passed away on dialysis. My mom was diagnosed with chronic kidney disease at 52 years of age, and my father in his 80s. So at first, my mom was told that she had polycystic kidney disease, and my father was told he had uncontrolled blood pressure and renal artery stenosis. So then when I was in college, I was tested for polycystic kidney disease and I was told I didn't have it. So, I went along my merry way until about 2019, I had lab works come back. And my EGFR was 72, which is still above normal. But I was like, oh, that's a little strange. Like, I'm a pretty healthy person. So I talked to my primary care doctor and he ordered lots of labs and a renal and vascular ultrasound. And everything looked good. I had like one tiny cyst. He's like, I don't think you have polycystic kidney disease. So of course I just, went about my merry way again. And then in 2022, after seeing more recent lab work where my EGFR continued to decline while my creatinine increased, I started getting a little bit nervous. So I took it upon myself to schedule an appointment with a nephrologist. Very nice doctor, ordered tons of labs, more ultrasounds, and he was thinking that maybe it was because I was severely anemic with borderline hypertension. So he added medication, come back in 6 months. So unfortunately, he left the office and moved to a different state. So they switched me over to a new provider who was very nice, but he was very focused on blood pressure and getting a biopsy. And so I even had one scheduled, but I canceled it a few days before because I was very concerned with complications as I've had like bleeding issues in the past. And so at this point, I start getting super neurotic and checking my BP, which like numerous times a day. Many times, I had told these doctors, both my parents were on dialysis. I really want to avoid that. So moving forward to this past summer, my numbers are still decreasing. I'm getting tired all the time. I'm like, gosh, this is so strange. And they just keep saying, come back in 6 months. I'm like, okay. So I work at university hospitals in Cleveland. So I asked one of my doctors who also has kidney issues, and he said, I know the perfect doctor for you. And so I had to wait 6 months to get an appointment with her, which just brings us to this past January. So this whole thing started in 2019, and now we're in 2025. So she was a wonderful doctor. She really listened to my concerns. And the first thing she said was, we need to do genetic testing. And I was like, oh my gosh, I've asked about that, but they just kind of were like, we need a biopsy. We need a biopsy. And I was like, I really don't want to do a biopsy. Anyway, so she had the box for me to take home, and I completed and mailed the kit the next day. So 3 weeks later, I finally had the diagnosis of UMON. So I pretty much read every single article written, and thankfully I found the Rare Kidney Disease Foundation, and Rich and Sharon have been wonderful. And it's not the result that I was hoping for, but I'm glad I finally have a diagnosis and an amazing resource from the RKDF. So I look forward to volunteering my time and helping others in my shoes. And really, I want like nephrologists to understand like genetic testing, like my insurance covered it. I didn't pay anything. And then I wondered throughout this whole why not one doctor mentioned genetic testing with both of my parents having kidney disease. So I calculated a couple days ago, but since 2019, I have spent almost $20,000 in lab work, ultrasounds, and appointments with specialists when maybe I could have been tested and just known and then like prepared myself.
Oh, Bonnie, thank you for sharing your journey and that you can still have a smile on your face. Uh, I'm just really impressed and thank you for joining us today.
Yes, thank you.
Stick with us. We do have some questions for you, but next I'd like to introduce Angela Watt. Angela is a retired renal nurse and a trustee with Kidney Research UK, the largest charity in the UK that funds research focused on the prevention, treatment, and management of kidney disease. Angela is also a UMOD patient. Thanks for joining us, Angela.
Thank you very much. And good morning, everybody. Good afternoon from the other side of the pond. Thank you for inviting me to speak to you today to talk about mine and my family's journey with kidney disease. So I can trace the kidney disease in my family over 5 generations. I know that my great-grandmother died of uremia and nephritis in the 1930s before dialysis was an option. My grandmother and her sister both had kidney failure and had transplants, one successful, one not successful. My own mother developed kidney failure in 2000, so I knew it ran in the family, but we never had a diagnosis. And then in 2006, my youngest son, who was a year old, was diagnosed with CKD. So that was the point when I realized I wasn't going to escape it. It was probably coming my way at some point, but we still didn't have a diagnosis. And it was only in around 2014 when I was eventually diagnosed with CKD that we ended up in the system, and I met with Professor John Sear, who is on the call, who I think you might be going to hear from at some point today. And luckily, John is a nephrologist at my local hospital who specializes in inherited kidney diseases. So he was the one who got the genetic testing done on myself and my son. Originally, it was through a project here in the UK called the 100,000 Genomes Project, which looked to map the genomes of 100,000 people with rare diseases where they didn't have a genetic diagnosis. And actually, they didn't find anything in it. They came back saying they couldn't find anything. And it was John himself who found it. He got the raw data back and had a look. And it was during the first lockdown, right at the height of COVID, he messaged me and said, I found something, I need to speak to you. And that was the first time I heard about ADTKD and UMOD, which is the mutation in our family. So since then, I have now developed CKD. I am now just about to start on dialysis myself. My son has had a successful transplant from my husband. He's doing really well. I have 2 other children. They have both been tested and they both do carry the gene. So the 50-50 doesn't work in our family. All 3 of my children have got it. And we have also found other family members. My brother carries the gene. He's now got CKD. My great auntie we knew had family. I was in contact with one of her children. And he carried the gene, although he didn't have the CKD. He had the juvenile gout that you get. But his daughter carries the gene and she has CKD without the gout. So Holly Mabillard, again, who you're going to hear from today is doing a lot of work into that, why there's that genetic difference within our family. Very briefly, by background, I'm also a nurse. I actually spent 20 years working as a kidney nurse. Probably the family history that made me have an interest in kidney disease, predominantly working in the hemodialysis setting. But in the last few years, I worked as a specialist nurse here in the UK for patients with another genetically inherited rare kidney disease called AHUS. And I've seen their journey and the journey we want to be on, where they started out with just a genetic diagnosis and now they have a treatment for it. That has stopped the kidney failure in their families and allows people to have transplants and things like that. And I've seen the difference it makes to them. And I want that for future generations of my family as well. So thank you. I'm looking forward to hearing what everybody's got to say today.
Thank you so much, Angela, for sharing your story as well. Let's see, Kathy, do we have questions for Angela or Vonnie?
Yes, we have a question for Vonnie from Tim. He wanted to know why you didn't want the biopsy.
Increased risk of bleeding complications. I had 2 other surgeries where I did have complications, and biopsies are not, there is risk involved to them. And even when you sign the consent, it says you may die. And I was like, I don't want to spend a week in the ICU with the bleeding complication. And that's one thing I feel that they don't really tell you. And there are a lot of biopsies that go extremely well, 99% of them. But there is the 1% that do have complications, and it also increases more scarring in the kidneys.
And I think, as we know, the biopsy is not useful in determining whether or not you have ADTKD. So it was very smart that you didn't have the biopsy.
Right. Yes.
Yes. Then we have a question, I think it's from Dr. Sayer for Angela. Let me just read. I think he wanted to know if a biopsy was ever mentioned to you, Angela.
No, it wasn't really, but I don't know, maybe because I was still in the early stages of my CKD, I don't know if I had progressed further without a genetic diagnosis, whether at some point they might have said, we should do a biopsy to see if we can find out what's going on. I think once we had a genetic diagnosis, that was it, pretty much locked in, and there was no need for a biopsy.
And we have one more question from Kelly for Angela. She was just wondering, how old were your kids when they had the genetic testing done?
My elder 2 children were both adults, so over the age of 18, I can't say exactly what, but old enough to make the decision for themselves. My son was still a child when he was entered with me, but actually it turns out that he has 2 rare kidney diseases. So his actual kidney failure that he required the transplant for, I think, was probably due to he had a grade 4 reflux. And that's what caused his CKD. And then the ADTKD finding was another anomaly sort of thing. So he has both of them, but it's actually the reflux that caused his kidney failure.
Thank you. That's all the questions we have for now.
Thank you, Kathy. And I just want to mention that Dr. Say also put in a comment to us that he really favors the idea of genetic testing prior to biopsy, not only because it saves all the pain and expense, but I'm guessing it's because it's less invasive, it's more conclusive, and then as you mentioned, Kathy, you can't diagnose ADTKD with a biopsy. Only a genetic test will detect that. And I'm sure there are other kidney genetic diseases for which a biopsy won't tell you any more information. So it's fairly invasive as a diagnostic tool. In many cases, it's warranted. I'm not saying it's not, but even Dr. Sayer is suggesting probably genetic testing prior to biopsy. So thank you for that, Dr. Sayer. Okay, it's time to move on to our next session. Thank you so much, Vonnie and Angela.
Thank you.
And next up, we have Holly Mavillard, and she's going to present an overview of ADTKD from both a clinical and a genetic perspective. Holly is a kidney doctor and a researcher based in the north of England, currently finishing a PhD focused on understanding why some people with ADTKD UMOD experience faster or slower disease progression, as Angela mentioned. Her work aims to uncover new insights about ADTKD that could lead to better treatments and outcomes for patients. Dr. Mavillard.
Thank you, Sharon. Can you hear me okay?
Yes.
Great. So thank you very much for that so far. It's been really interesting to hear from Vonnie and Angela. And really, Sharon has given me, was asked me to give you a very quick whistle-stop tour of ADTKD, really from the clinical and genetic perspective to kind of set the stage for discussions today. So I hope this is helpful and informative to many of you. So first of all, what is ADTKD? So autosomal dominant tubular interstitial kidney disease is a clinical entity, and it's defined by interstitial fibrosis of the kidney and tubular damage. Also bland urinalysis, so the absence of blood and protein in most cases on the urine dipstick, and progressive kidney disease. And as you can see here on the slide, it's one of the 3 most prevalent genetic kidney diseases. And this is after, certainly in the UK, Alport syndrome and ADPKD, polycystic kidney disease. And it probably accounts for about 5% of all monogenic causes of chronic kidney disease. And as many of you will know, because the disease was formerly known under a variety of terms, so medullary cystic kidney disease or familial juvenile hyperuricemic nephropathy and other terms, this has really hindered recognition and accurate diagnosis for patients and their families over the years. So now we refer to this unified term ADTKD, which I think is much better. And the true prevalence of ADTKD is largely unknown. And this is for a couple of reasons. This is mainly because of this limited clinical recognition for a variety of reasons, but also difficulties in diagnosing one of the most common subtypes, ADTKD MUC1, which we'll touch on. And as some of you will know, the disease in its 2 most common terms, if you look on the right-hand side, ADTKD UMOD and ADTKD MUC1, are limited to a very small portion of the nephron in the kidney. So in the case of ADTKD UMOD, limited to the thick ascending limb of the loop of Henle, and also in the distal nephron when it comes to ADTKD MUC1. And what happens is there's a mutation in either one of these genes, which involves a toxic gain of function effect, which leads to intracellular accumulation of a misfolded form of the protein, either uromodulin or mucin 1 proteins. And what this does is it triggers endoplasmic reticulum stress and ultimately leads to interstitial fibrosis, which some people may recognize as a type of scarring to the kidney. So I'm going to talk you through the subtypes for those of you that are less familiar with this. So given the non-specific presentation of ADTKD, molecular genetics, as we've just heard, play a really important role in diagnostics and classification of the disease. And this is reflected now in the terminology where the causal gene is appended to ADTKD, as you can see in the diagram here. And to date, there are 6 genetically distinct subtypes shown on the slide. ADTKD UMOD is the most common subtype, followed by ADTKD MUC1. And the 4 other subtypes are much rarer and usually present with more extrarenal manifestations, as you can see in the coloured boxes. Very, very rarely, mitochondrial gene variants or mutations can also cause the disease, and we call this MITKD, or mitochondrially inherited tubular interstitial kidney disease. And obviously the familial pattern is a bit different in that situation. My most important point here is that a lack of genetic diagnosis does not rule out ADTKD even after you've tested for these genes that I've mentioned. And this is because we still believe that there are genes causing the condition that are yet to be characterized or discovered. And we call this ADTKD NOS, or not otherwise specified. So how does it present? Some of you may be more familiar with this than others. But typically and classically, particularly in its most common forms, ADTKD, UMOD and MUC1, there are no defining clinical features really. You might have absence of blood or protein on the urine dipstick, and the kidneys on imaging such as ultrasound might look normal in size and structure. Many of these patients, consequentially, that we see in clinic are either diagnosed because a family member has been diagnosed and we've tested them, or on a random blood test, they've been found, as we've already heard, to have a low GFR, a lower kidney function, and that's prompted us to consider what the cause might be. So even though ADTKD is not primarily a glomerular disease, we do in rare situations see it present with protein in the urine. And if a biopsy is done, we might see a pattern called FSGS. And this can be a presenting feature in more advanced forms of the disease. And the reason I mention this is nephrologists out there should always have that diagnosis of ADTKD UMOD in the back of their mind in cases of secondary FSGS where there is more than one person in the family affected or there's a genetic flavour. So when I learned about ADTKD initially, the hallmark presentation in the textbooks was always of adolescent onset gout. And I wanted to highlight that this isn't always the case, but it is still a hallmark of the most common form, ADTKD UMOD. And in a European cohort, about 50% of women and 75% of men had gout. Now, this can be adolescent onset or under the age of 40, but not all patients get it. And in the case of ADTKD MUC1, gout can occur, but it's less common and occurs at much later onset. In ADTKD UMOD, a third of patients get kidney cysts. So people might get the diagnosis of cystic kidney disease. But these are small cysts and typically don't expand the size of the kidney. And in rarer and atypical subtypes, you get more extrarenal features. So on the right-hand side, this is a lovely diagram reflecting ADTKD, HNF1 beta, and the variety of presentations that you can see clinically outside of the kidney as well. And again, my most important takeaway from this slide for clinicians and patients is that ADTKD is extremely heterogeneous. And what I mean by that is the disease looks very different often within a family where the same mutational variant is causing the disease. And we've got families where the onset of kidney failure, even between brother or sister in the same generation, can differ by over 30 years. And that's very much what my research is trying to understand. So how do we diagnose it? And we touched on this before. So typically we are looking in the most common forms for a lack of discriminatory clinical findings. And then family history is often positive. I would encourage you to look for an autosomal dominant pattern of inheritance. Of course, molecular genetic testing, and this is, as we said, the gold standard for diagnosis. And Vonnie touched on this, kidney biopsy is usually non-specific anyway, so it's much less informative. In terms of genetic testing, and I know further presentations are going to expand on this in much more detail, but just briefly, next generation sequencing is the standard for genetic testing. But of course, many of you know that due to the variable non-tandem repeat region of the MUC1 gene, variants are not detectable via next generation sequencing. And so we require specialist methods such as immunohistochemistry and a newer method called VN-typer in order to diagnose this subtype. And these are available via the Broad Institute at Harvard, as you mentioned before, Sharon, and also ADTK-NET Consortium and academic institutions in Europe. And I really wanted to show this slide because I really hear these things all the time in my clinical practice. ADTKD is plagued by common misconceptions, in my opinion. And I've touched on a few of these that I hear regularly. Slide. So firstly, in terms of when people say, why bother to test this disease when there are no treatments? Well, if we don't have people with the disease to study, we're never going to get closer to treatments in the first place. And if we don't test for it, this results in missed opportunities for not just future treatments and clinical trials, but genetic counselling, access to prenatal diagnosis and counselling as well, and of course precious research involvement that I have certainly relied on so heavily from patients around the world. As I mentioned before, not all patients present with juvenile onset gout. So that textbook clinical finding that I was taught at the start of my doctor training is not always present within patients and their families. De novo cases where the mutation occurs for the first time in a person or a family can occur in all of these subtypes. And when people tell me, but it's very, very rare, the way that I think about this is, as a kidney doctor, when you walk onto a kidney unit, you've got a high concentrations of patients with chronic kidney disease and kidney failure. And so ADTKD is going to be much more prevalent in this setting, and we must not let CKD of unknown cause go unexplained. So we must strive to genetically test these patients if we can't get if we don't know what the cause is. Negative genetic testing excludes a diagnosis. Well, I hope I've explained to you regarding MUC1 and ADTKD NOS that's not always the case. And one thing that I want to emphasize here is that certainly in the UK, I'm not sure about the extent in other countries, but we have amazing access to pre-implantation genetic diagnosis, where patients can reduce the chances of this disease being passed on to future generations. And I make a point of explaining this and offering this form of preconception counselling to all people who have not yet had a family or completed a family, whether they're a man or a woman. And of course, we're going to hear more about the hope on the horizon for future treatments that are coming. And so to detail a bit about the landscape, to contextualise the condition, at the moment, the majority of cases are reported in North America and Europe, and this probably reflects access to diagnostics rather than geographical restriction, and we hope to expand this more globally. Awareness, of course, is growing elsewhere, and improved genetic testing is, of course, helping. But one interesting aspect that you might be keen to hear about is that in the UK, we've got a unique mutation causing ADTKD UMOD, which is extremely common and accounts for more than 50% of cases. And this subtype presents mostly without gout. Most of our patients don't have gout, and so it's even harder to clinically think about ADTKD UMOD in these cases because you don't have that hallmark sign that provides brilliant opportunity for researching the disease without the confounding effect of different UMOD variants. And of course, registries and consortia helping to map the international landscape. And I've illustrated just a few, mostly in Europe and North America, on the right-hand side here. And just to touch on the current gaps in knowledge and research efforts, to give you an overview, we're very much still focusing on characterizing genotype-phenotype correlations in all forms of these subtypes. Certainly my research is very much focusing on finding genetic modifiers of disease progression. I think we'll hear about disease biomarkers a bit later in the summit, and my research is also involved in this. We're looking into protein misfolding and that cell stress response and understanding that better. And of course, disease-specific therapies that we'll hear more about later. And of course, we need research modules that really mimic that human phenotype so we can test treatments and get them into clinical practice quickly. And so, from the perspective of a kidney clinician and a researcher in this disease, I've just written a few key takeaways here that I want to get across. And my main one is that misdiagnosis and underdiagnosis is extremely common with this condition. And outside of the specialist setting, if you're in a renal unit without someone with a specialist interest in genetic kidney disease, the patient and their family may need to prompt their nephrologist to consider this as a diagnosis. And I would encourage anyone to get in touch with people on this call or with the charitable organisations to help, and we can signpost you to people that can get you a diagnosis. Of course, international recognition and collaboration is essential, and I've very much relied on my collaborators to help move our research forward. And we need this not just to pool data, but identify trends and raise global awareness. And intense research efforts are ongoing. One thing that's interesting about ADTKD is that it's a monogenic model of kidney fibrosis. And so treatments in this disease may well be relevant for other kind of a wider spectrum of other kidney diseases, which at the end point is interstitial fibrosis. So there may be very broad implications for research and treatments in ADTKD. And we're getting closer to mechanistic therapies, and I'm very excited to hear more about that later in the summit. So thank you for listening, and I hope that was helpful.
Thank you, Holly. That was wonderful. That was a great overview. And I even learned some things that I hadn't known previously. So thank you very much. Kathy, do we have questions for Holly?
Yes, we have a few questions. The first one is from James. He has UMOD, but he also has severe proteinuria. And as we all know, we thought that that was not usually associated with UMOD. Is that possible to have that with UMOD, Holly?
Absolutely. And so I wouldn't discount if someone has proteinuria, whether it's mild or what we call subnephrotic. I wouldn't discount that. And as I mentioned at the start, so there's we do see cases of ADTKD UMOD presenting with something called focal segmental glomerular sclerosis. In those settings, I would expect a kidney biopsy to often be done, but obviously if you can get a diagnosis via genetic testing, that is better for the reasons that we mentioned. This typically presents with more severe disease, and I would expect that antiproteinuric drugs that we have access to now would be given to the patient as well, to help slow down the disease progression there.
Thank you. We have a question from David. In your research, have you found any causality for significant variations in clinical courses for siblings?
Yeah, so that's very much what my research is focusing on. So I've looked at DNA samples from over 600 patients with ADTKD UMOD, thanks to the, largely to the help of Tony and Kendra and other collaborators around the world, and we're looking to expand on this. And at the moment, we've got some interesting leads, which I'm hoping to share amongst the community and publish in the coming months. One of them is a link to epigenetic factors that control kidney processes. The second is a link to the mitochondria and the biology around that, which we already know a little bit about from research around the world. And the final interesting thing that's come across from the research is that genes involved in uric acid transport in the kidney have all come up as genes of interest that may be modifying kidney disease progression. And also polygenic factors. So that means in our background DNA, we've got lots and lots of genes that have little tiny individual effects, and when combined have a kind of more magnified effect on disease progression. And so to kind of summarize, those are the interesting leads that I've got at the moment, and I'm hoping to validate this thanks to the ADTKD Net Consortium. And so we're working really closely around the world to try to really prove this in more detail, and then to follow this eventually, I imagine, will be experimental models.
We have a question from Dr. Sayer. How do we get other doctors and healthcare specialists interested in kidney genetics and ADTKD?
That's a very good question, John. I think the tagline, and Angela Watts often said this to me, and I totally agree with her. The tagline of never letting chronic kidney disease of unknown cause be allowed, unless you've got a genetic or a histological diagnosis for your kidney disease, you cannot make assumptions. And from my personal experience, a lot of these patients we've re-diagnosed with ADTKD UMOD, and they've had for years in the family a kind of presumptive diagnosis of hypertensive nephropathy, or like blood pressure induced kidney damage. And so I think really, certainly from my perspective, I'm trying to get that message out there as much as possible by presenting around the world and presenting at conferences and educating nephrologists. So I think we can all do that in our own ways, and this summit is certainly a brilliant way of getting that message across.
Thank you so much. That's all the questions we have for now, Sharon.
Thank you.
Great. Thank you so much, Holly. I appreciate the presentation and your questions. And if folks have other questions for Holly later in the program, put them in the Q&A box, because we will get to them. So once again, thank you so much for joining us and for that fantastic presentation. All right, moving on. Next, we have Dr. Guillaume Dorval, who will share the latest advances in genetic testing for ADTKD. Dr. Dorval is a pediatric nephrologist and assistant professor of genetics at the University of Paris. His research focuses on understanding the molecular mechanisms behind glomerular kidney diseases, as well as developing innovative therapeutics. His work on the MUC1 gene has led to the development of a freely available analysis tool, VN Typer, which he will be discussing in his talk. And I'm happy to share your slides if you'd like, Dr. Dorval, or you can go ahead, whichever you prefer.
Well, you can share it if you have.
Okay.
Thank you very much. Thank you for this nice introduction and for the invitation to talk today at the ADTKD Summit. I think I will shut off my camera because I'm not sure that I have enough network for that. But so I'm very happy to be here just to talk about the advances in genetic testing for ADTKD, and especially for work on MUC1 ADTKD. On the next slide, please. So I will be very brief in the introduction, since Holly did a very complete overview on ADTKD and different genes involved in the ADTKD. I think that one of the most important thing is that what she said, ADTKD is not a very specific disease, and we don't have any specific clinical phenotype for ADTKD. And we have a genetic heterogeneity with at least 6, but now more genes involved in ADTKD, and we will find probably more genes to be discovered in the next years. The next slides. So 10 years ago, the KDIGO, so the recommendation suggested that gene testing and demonstration of a pathogenic variation in one gene involved in ADTKD is necessary to establish the diagnosis. So it's of utmost importance, since patients with ADTKD can visit at the clinic without any family history. And so they visit as a sporadic case, and TKD diagnosis may be challenging in such patients. So the place of the genetic role for those patients is very important. In the next slide, the question is how to test these patients. So gene testing evolved a lot since 10 years. Third generation sequencing is now more and more available and will probably be again more available in the next years. But unsupervised gene testing started with the second generation sequencing with massive and parallel sequencing of panel of genes, exome, or even genome sequencing since 2010 about. But in the recent years, third generation sequencing allows the sequencing of single molecule that is very important for very difficult to sequence regions, such as what we call GC-rich regions or long repeats and so on. And you will see that in the very important for the MUC1 gene. On the next slide, please. For many genes involved in ADTKD, gene testing is now easy because there is no technical challenge for diagnosis mutation. I mean, just for adding sequencing genes and not for filter variants, but for the sequencing step for UMOD, REN, HNF1 beta, SEC61A1. And so we have algorithm for patients and we can detect mutations with regular sequencing. But for MUC1 gene, it's very, very challenging because of its very complex architecture. On the next one. So MUC1, you know this protein. This protein is expressed in the distal tubule. And in the next one, it's encoded. The protein has a very long extracellular domain. And this extracellular domain on the next slide is encoded by a very, very specific region that we call the VNTR. VNTR is for variable number tandem repeats that consist of 20 to 120 repeats of 60 base pairs with a very high homologies, more than 95% of homologies, and you will see later. On the next slide, we know that some specific mutations in the MUC1 gene lead to what we call a frame shift, so a change in the sequence of the gene, of the mRNA, and this leads to the synthesis of a neoprotein, and this neoprotein can be stained in cells and is very toxic for cells and leads to ADTKD in patients. The next. And so due to its very specific architecture, sequencing this region is a nightmare for geneticists. Schematically, we can find at least 4 or 5 factors that make the genetic diagnosis highly challenging. The first one is that it's because of the VNTR. It ranges from 20 to 120, 60 base pair repeats, and it's really difficult to amplify it. So technically, it's really difficult to just amplify this gene. The second is that pathogenic variants are usually located in longer repeats of longer MUC1 alleles. And due to technical bias, it's difficult to PCR to amplify these alleles. The third is that the structure of the gene is composed of a lot of, like we call GC, and it's really, really difficult to sequence this kind of genetic structure, and it leads to sequencing errors that can mimic mutation. So it's really difficult to make a difference between a sequencing error and a true mutation in patients. On the next slide, you can see that when you sequence a patient with what we regularly use, that is a short read sequencing, so it's 120 base pair sequencing, due to the repeats and the variable repeats in patients, you have like a dilution effect and you can't detect the mutation easily, what you can do in other genes, and you have misalignment of reads in of sequences in these genetic regions. And so on the next slide, in 2023, with Hassan Sey, who is a PhD student, who was a PhD student, now a postdoc, in the lab, we developed a bioinformatic tool that we call VNTyper, and only talk about this tool. And this tool can use a very specific bioinformatic structure just to try to identify mutation in this gene from short-read sequences, so from regular sequences of patients, what everybody can do now. On the next slide, very schematically, the ID, I won't go into details, but the ID is to predict the sequence of the patients based on all reads from the patients. And so it's like a prediction. And when you predict the sequence, you can after compare the sequence to all possibilities of VNTR that have been published so far and detect one region that is different from the reference, so the VNTR that are published to date. And on the next slide, from a clinical point of view, we validated this tool on an ADTKD cohort, so in patients for whom we knew that they carried a pathogenic variant in MUC1, and we re-identified this pathogenic variant in 96 patients. And on the next slide, we used after that a huge cohort just to try to identify new patients. And we were able to identify 30 new ADTKD patients. On the next slide. What was very interesting with this tool is that for the first time, it was possible to do unsupervised MUC1 analysis. Why is it important? Because before you could only test MUC1 gene if you thought that, if the clinician thought that the disease could be due to MUC1 gene, but it was not possible to test the gene in a patient if you don't think about MUC1 related ADTKD. And on the next slide, when we tested the whole cohort, you can see that, of course, we detected MUC1 mutation, mostly in patients referred to our center for ADTKD, but we detected also mutations, MUC1 mutations, the regular DUP-C mutation, but also other kind of mutations in patients without any ADTKD, for example, phenotype of nephroptosis or proteinuria, glomerular proteinuria, or like KQT, and I will show you after why. On the next slide, we tested again this tool on the larger cohort in the lab, and we detected again 33 new index cases in the cohort. And interestingly, for 40% of patients, ADTKD was not suspected by the clinicians who referred the patient to our center. And on the next slide, we detected 9 new variations. You know that usually in this gene, we have a highly prevalent variation that is a DUP-C variation in the repeats of 7C. And we detected new variation leading to the same kind of phenotype. And in the next slide, this work has been replicated in other cohorts. For example, in this one, in a Finland cohort of 123 patients, and they detected 5 MUC1 with regular mutation, regular DUP-C mutation, and 2 other mutations that are not reachable by the standard gene testing for MUC1. On the next slide, if we just go a few seconds on patients in whom ADTKD was not suspected, what we can see is that the age at diagnosis and also the severity of the disease was higher in these patients. And of course, we didn't find any positive family history in 6 out of 11 of patients in whom we did not detect ADTKD, while we didn't thought about ADTKD, sorry, but family history was found in 95% of patients in whom ADTKD was suspected. The next slide. And when we just focus on patients without any family history, it was possible to confirm the de novo mutation in 2 patients, explaining the absence of family history in such patients. And you can see that in both patients, the disease was severe, with kidney failure at 32 and 28 years old in both. And in 2 other families, we detected a MUC1 mutation that was hidden by another mutation, and we found 2 mutations, one deletion in HNF1 beta and one mutation in IFT 140 linked to cysts in these patients. On the next slide. So the main question now is it possible to adapt this tool to all exome sequencing data? And what we can see here is that when we try to delete some reads, so to decrease the read depths in patients with high read depths at the early beginning, we can see that in green, you can see positive diagnosis in true positive patients. In orange, it's in between, so it's really difficult to answer. And in red, Ventyper failed to detect the mutation. So what you can observe is that this tool is highly sensitive to read coverage. And so it's really, really important when the clinician, the geneticist perform a gene testing to check for the read depths and to check for the tests. And on the next slide, the read depth is highly dependent on the technology that is used by the lab. And so it's really important also. I compared here 2 technologies. We can compare more technologies, of course, if we want, but you can see that for one technology, you have a good read depth in this specific region, but with another technology, you fail to have a sufficient read depth, and so you fail to detect mutations in patients. And there is one French paper that I don't like very much, but that identified 32 patients by exome sequencing, and out of more than 3,000 patients, but we don't have any data of read depths, phenotypes, and so on. The next slide. And in a joint effort, Asensei and Bernd Popp from Berlin, they developed this tool on a web server, so the online tool. And so it's a freely available tool for everybody. So you can test your patients through this tool, and you can test either a patient or a larger cohort of patients. And so do not hesitate just to go to test some patients. I regularly receive emails from clinicians or from geneticists just to try to test for patients. The next slide. Sorry. Yeah. We can't talk about MUC1 gene without talking about long reads, because long read is really a gold standard to detect mutation. As compared to short read can just read genes by sequences of 100, 120 base pairs, whereas long reads, depending on the technology, can sequence several thousands of base pairs in the same during the same time. And so you can detect you can sequence all the VNTR in a single, what we call a single molecule real-time PCR. On the next slide. And so there are some papers, and some very beautiful papers that use this kind of technique. This technique is beautiful and is a gold standard for validating mutation is a MUC1 and to localize the mutations in the VNTR. But this technique is a supervised test, since you must do a PCR, so an amplification of MUC1. So the clinician must think about MUC1 before doing these tests. And as far as I know, there is no like whole genome sequencing, not yet, but they will have publication in the next weeks or months, of course, with whole genome sequencing with PacBio or any technology. On the next slide. So as a conclusion, what we can say is that ADTKD causal genes, all ADTKD causal genes can be sequenced by next generation sequencing. So by regular sequencing, Ventyper enables the unsupervised sequencing of the MUC1 gene, and this allows identification of patients with atypical phenotypes, and this is very important again, just for clinicians and for patients, and to identify new variations different from the DUPC that was very difficult to identify before. Study of large cohorts are in progress, and in particular from exome sequencing, and long-read sequencing remains the gold standard because VENTAPER is coverage dependent, and so we should confirm some mutations in case of low precision by VENTAPER. We should confirm mutations by long-read or by another technology. And of course, this sentence was true in the CADIGOs in 2015, and this sentence is again true today, that failure to identify mutation does not exclude the diagnosis of ADTKD, and not all pathogenic genes have yet been identified. I thank you all for listening, and of course I thank all my lab and all our collaborators for this work. No, I can't hear you.
Thank you.
Ah.
Thank you for that wonderful presentation. And Kathy, do we have any questions?
We have time for 2 questions. The first one is from David. He wants to know which labs in the world can perform this advanced form of sequencing, as this would be useful for patients who have an unidentified mutation.
For which kind of sequencing? For the VENTAPER or any sequencing?
It just says advanced form of sequencing. I'm not sure.
For the VENTAPER, you start. It's really a bioinformatic tool, and so you can start from any sequences from any lab, and now any lab can adapt and can test the patient sequences in the website. And of course, we can help for anything if we need if the help is needed. Regarding long-read sequencing, not all lab can do long-read sequencing. I don't know whose lab can do it. I know that Martina Zvina in Europe can do it. Of course, I believe that Broad Institute do it, long-read sequencing, but not all lab can do long-read sequencing actually.
Okay, thank you. And then we have a 2-part question from Dr. Sayer. When you find more than one genetic variant, how do you decide the weight of each variant and their impact on disease? Can VIN-type be applied to WGS data as the read depth is much lower? And he also asks, is eyeballing the BAM files in the region a possible way to look, or is it messy even in controls?
For the first part of the question, we found it in 2 patients, to be honest. The first one was a 3-year-old patient with HNF1 beta deletion, and so we knew that the MUC1 didn't have any role in this patient at this time, and the parents didn't want to test themselves, and so we don't know. And we don't know if the patients are symptomatic or no, because they decline any gene testing, but also any urine tests. So we don't know anything about patients. So it's really difficult to decipher between the 2 mutations. In the second patient, it was a DUPC insertion for the DUPC in the stretch of 7C, and what we found, it was associated to an IFT 140 mutation in a quite early, a young patient, and this patient had a kidney failure with the IFT, and so I think that both genes can have a role in the disease, the IFT with the cyst and the MUC1 with the mucoin in the disease course and the kidney failure. The second question of the second part of the question was, is VN-Typer adapted to whole genome sequencing data? To be honest, actually, I don't think that it's possible, because usually we have like 40, 50 read depths in whole genome sequencing data. We have to check what kind of read depths we have on the specific region of VN-Typer, of the VNTR, but I'm not sure that actually we can adapt it to WGS, and we try to adapt it, and we try to to limit the failure in all exome data. And there was a third part, I think, but I missed it.
I think that's all the time we have for questions, and I think that was both parts of Dr.
Two
Sayers.
more questions came back, but I think we have to move on, so.
Okay.
Okay.
So a lot of interest, obviously, in this free online VN-Typer. So thank you very much, Dr. Dorval.
Thank you.
Next, I'd like to introduce Dr. John Sayer, who will be moderating this session. Dr. Sayer is a professor of, this is not Dr. Sayer, but she will be in this session. Here he comes. Thank you. Dr. Sayer is a professor of renal medicine at Newcastle University in the UK. He has been an academic nephrologist for 21 years and specializes in studying genetics and molecular mechanisms underlying renal diseases. And John will introduce Gila. So welcome, John. Thank you for joining us.
Hi, thank you, Sharon. Thanks, everybody. Really interesting talk so far, and I've got the pleasure of introducing the next speaker. So this is Dr. Gila Razuli, who's an assistant professor of medical sciences and associate director of education at the Center for Precision Medicine and Genomics at Columbia University. So welcome, Gila. Thanks moderating this session. I've got the easy part, you've got the more difficult part of giving us the talk, but
I have
great
the
to have
easy
you
part
with us.
because everybody else already covered, I think, almost everything I have to talk about. So it's going to be a review of the prior talks. Okay, so I'm going to talk about genetic testing in clinical practice and try to answer three questions: why, when, and how. So I think that Vonnie and Angela really explained the why, and Holly also had a really nice slide explaining why. And the how, the technical side of the how was reviewed by Guillaume, and I will try to talk more about how genetic counselors can help there, and how not everybody needs to become specialized in genetics. So why offering genetic evaluation to individuals with kidney diseases in general? I think, again, it was well covered, but there are all kinds of motivations. Some can be seen more as patients' motivations, some may be more clinician motivations, but probably it's like a spectrum. So it can create an informed and appropriate treatment plan. It can lead to appropriate extrarenal surveillance. It can determine eligibility for clinical trials, as was discussed earlier. It can obviously clarify the etiology of the condition, the why did we ever develop this kidney disease, elucidate the prognosis, what's going to happen to me, how fast I'm going to get on dialysis. It could provide access to reproductive planning option, as was discussed by Holly. Some places have more or less access to pre-implantation genetic diagnosis, but it is an option. It can help determine the risk of family members, which we call cascade testing, and was really nicely presented by Angela, who spoke about all her family members who were diagnosed following her diagnosis. Can provide access to disease community like this one. And it can end the diagnostic odyssey, which I think was, again, Vonnie really told us about her odyssey from 2019 to 2025. So I think these are all good reasons. There are probably many other reasons, and any motivation, any reason is a good reason to get genetic testing. Really, I think one of my main take-home points for nephrologists and other providers on the call is that you should not be the barrier between the patient and genetic testing. So even if you don't know how to do genetic testing, how to refer to genetic testing, that's something that you should know, how to refer to a specialist who will know what to do. So there are potential impacts of genetic diagnosis on actual clinical management. So again, going back to Holly's talk, there are things to do, even if there are no treatments now, they might be in the future. And it might inform kidney transplantation and the choice of the donor, if the donor is a family member. And it can also give access to family planning and to decisions around family planning. And as it was discussed, it might lead to new trials. So just knowing that you have a specific disease can make you eligible to a trial. And having more people eligible to trials increase the pace of these trials, because usually one of the most time-consuming part of a clinical trial is just to enroll patients. These are just three websites as examples. So the clinicaltrials.gov is usually for mostly phase 2 and 3 trials, but also phase 1 trials. The reporter.nih.gov is a website of all the research done and paid by the NIH. And these are just the 4 trials that research that are ongoing right now around ADTKD. And this is usually the step prior to the clinical trial, but it can also be the clinical trial itself, which is sponsored by the NIH. So clinical trials can be through NIH or through other organizations or through pharmaceutical, etc. And then in Europe, there is the Orphanet that is a good resource to find trials. So when should you offer a genetic evaluation, assuming that the patient is not asking for it, but you as the provider is the one who should suggest it to your patient? There are some red flags here that I'm going to highlight. They don't include the motivations that I mentioned earlier, but again, motivations are a good reason to offer genetic evaluation. So for instance, if you have a patient who doesn't have any of the red flags I'm going to cover, but is really worried about transmitting his or her kidney disease to her children, that's a good reason to refer to genetic testing. Okay, so some of the like well accepted red flags are, first and foremost, family history. So if your patient has a family history of kidney disease, which is relatively common, it is a good reason to refer to genetic evaluation. And I'm calling here genetic evaluation and not genetic testing because you may not be the one actually ordering the test, but you just want to refer to a geneticist or to a nephrologist specialized in genetics. An unusual clinical course in pink here is another reason to refer to genetic testing. If your patient doesn't look like your mainstream most common presentation. And any extra renal involvement, anything that is not related, not caused by the kidney disease, but something else like gout or any others. The things here are not so much about ADTKD, but we would think about like early onset diabetes, for instance, for the ADTKD HNF1 beta form. Certain kidney subtypes, and I want to highlight here the unknown etiology is a major reason. So if you don't know why your patient developed kidney disease, it's a really good reason to refer to genetic testing. And then if you have a concern about a specific genetic condition, for instance, if you suspect ADTKD, it's a good reason to refer to genetic testing for ADTKD. So again, you found your patient, you think they could benefit from genetic counseling, but you actually only learn very basic genetics through medical school, fellowship, and licensing, and you are in a generalist nephrology clinic. What should you do? You don't have to become a genetic expert. You can refer to geneticists or genetic counselors, or you can refer directly to centers of expertise, which I will highlight at my last slide. Or you can choose to become an expert in genetics through CME courses, workshops, or you may want to take even advanced training. And there are now subspecialties in Europe. There is I think a 1 or 2 years training for to become a nephrologist expert in rare diseases and genetics. And in the US, there are a few places that offer this type of advanced fellowship as well. So you may want, well, should I refer to genetic evaluation or should I order a test myself? In the case of ADTKD, and let's say in the case of the United States, you may say, well, let me first order the general test that I have, a broad panel. I'm not sure what my patient has. If the test comes back negative, as Holly mentioned, that does not mean that it's not genetic. It might have been missed. As both Holly and Guillaume highlighted, the MUC1 variants are missed by regular tests, and that's a bit more complex. And you may not feel comfortable actually reaching out to Tony Blair and order it and ask him to help you out. So that's when you may want to refer to a clinician with genetics expertise who might be a genetic counselor or not. If you also feel like there are reasons for risk of adverse psychological impact of having a genetic diagnosis for your patient, that might also be a reason to refer to genetic counseling as opposed to ordering the test yourself. So for those of you who want to do the genetic testing, order the genetic testing yourself, I want to highlight the 4 steps of genetic counseling. But for those of you who don't want and just want to identify your patient and then refer them, the first 2 steps are actually should be part of the nephrology regular clinic anyway. So collection of medical history, that's part of any meeting with any physician. In this case, we want to formulate the most specific kidney disease diagnosis. Again, if we cannot, that's a good sign that we should be referring our patient to genetic evaluation, evaluate for extrarenal features, determine the kidney disease age of onset and the rate of progression are also important things. And here in the white bubbles, you have some questions that you should probably ask yourself and then ask your patient. And then the second part is family history. And I'm going to go deeper into both medical history and family history in a second. And then there is the etiology assessment, which can be done by, again, a clinical geneticist or a genetic counselor or nephrologist. And feels comfortable with that, where you actually estimate the probability of a specific kidney disease. You're going to formulate a differential diagnosis and think about which test to order. And then there is the informed consent, which is an important part of ordering genetic testing for patients. We want to make sure that the patient makes an informed decision to do the test. They need to understand the test, its purpose, the potential results, their impact on management and family, and discuss the process of testing and returning results, and the potential of having variants of uncertain significance, the potential of having a negative, unconclusive result, and what would be the next step in this case. So in the case of ADTKD, for instance, if the broad test is offered as part of informed consent, there should be an explanation that there is a significant part of people with certain diseases that we need additional tests for MUC1 in the ADTKD case. Okay, so let's deep just in a little bit into the medical history. So again, these are the things that I already talked about. So you want to ask yourself, is the patient's progression of kidney disease typical? Is the age of the patient typical, or is it earlier? There is now kind of expert-based agreement of 50 years old, but there was a recent paper showing that after 50 years old, you also find a lot of patients with genetic causes. So it's really a combination of any of the signs that should make you think about genetic testing, not everything at once. What is the most likely clinical diagnosis? Does the patient has non-renal disorders? When I'm talking about non-renal disorders, it might be eye anomalies or blindness, hearing loss, heart anomalies, heart disease, polydactyly, having 6 fingers, obesity, diabetes. And these are just some examples that none of them is ADTKD, but you could think of gout as being the extra renal for ADTKD, which again, not having gout is not a reason not to think about ADTKD, but it could be a reason to think about ADTKD. On the family medical history, and it's really what differentiates between genetics and any other medical subspecialty is that family history plays a big part here. So we need to collect a 3-generation pedigree, 3-generation being including the grandparents, and as possible cousins. Understand whether there are other family members with kidney disease, are people passing away young. We may not know at a certain point that people passed away because they had ESRD that was not treated properly, but if they passed away young, then that's a sign as well. Are there other serious health conditions? Maybe another family member who's not diagnosed with the kidney disease, but something else that is associated with syndrome. Is there any possibility that the patient's parents could be related to each other? And that's relevant for recessive diseases and not for dominant diseases. So just as part of thinking of a genetic diagnosis, you need to become a detective. And so you should avoid asking just, is everyone well? Because patients would just say yes. You want to ask very specific questions, and you want to be systematic to ask about each person in the family. You should look at your patient's body language to understand whether they know or they don't know. Often patients say that they don't have, that someone doesn't have anything when they actually have no idea if they have or not. So it should be explained to patient that it's okay not to know. They should make a difference between the two. Be sure to record the age, onset of symptoms, and collect information on major medical, physical, or mental health problems. Anyone needed treatment in hospital, serious illness or operations, miscarriages, or babies who are stillborn. So some examples of questions: how many siblings do you have? So that you start with them, and then you make sure that you cover all the siblings. Did you have any siblings who passed away? Or does your mother, father, siblings, aunts, uncles, grandparents have kidney disease or any of the other issues that you want to cover? Now, I realize that that's a lot for a 10-minute clinician meeting. Part of these things can be automatized. There are now all kinds of tools to collect family history. Some of that can be completed by the patient first and then reviewed. And you can also have nurses or nurse practitioners do that with the patient. As a genetic counselor, I think that drawing a pedigree can really help being systematic and making sure that you collect all the family history. And that's just to show you how we would look at that. So most commonly, people with a recessive disease don't have any family history, even though in textbooks we're always shown people with related parents and like highly interrelated family members. So there are multiple people, or they have multiple siblings. But since most of us don't have many siblings, and most of us don't have families or parents who are related, the most common form when it's recessive is that there is absolutely no family history. Similarly, in autosomal dominant, often there is no family history, either because it's de novo, it happened in your patient, DNA and it's absent in the parents, or because the parents didn't share information with their kids, passed away for other reasons like accidents. But I would say that not sharing information between family members is probably the main reason why there is no family history. And then in the X-linked, which means that the variant, the gene that is causing the disease is on the X chromosome, usually women don't have a presentation, even though in the recent years we see that more and more women have just a little bit of a different and less severe presentation. So my main takeaway point is this slide. You should refer to a genetic counselor if you're not sure, if you think your patient may benefit from genetic counseling and genetic testing. Genetic counseling doesn't mean that they have to have genetic testing at the end. This discussion that they're going to have with the genetic counselor, and then at the end, in informed consent, the patient is going to decide whether they want to get genetic testing or not. In the United States, there is a website called Find a Genetic Counselor that is provided by the National Society of Genetic Counselors, the NSGC. There is in-person and telehealth options. And when you're clicking on this website, you can actually go specifically to find genetic counselors who are specialized in nephrology. And going back to Orphanet, which is covering all the countries that you see here, so mostly Europe, but not only, there are expert centers, and so you can enter, and Orphanet is for orphan diseases, so rare diseases, and you can find experts in kidney diseases, or specifically even more for ADTKD. So I think I'm going to finish here. I'd like to thank you and thanks all my colleagues, and feel free to reach out to me if you have questions.
Thanks very much. So I'll start some questions while we're waiting for people to post their questions, but that was a really great overview. But I think we are all passionate about genetics and studying this disease, but I want you to answer some questions on maybe the negative sides of genetic testing, or the preconceptions about negative sides. So one is, like, when the sample's sent away, there's often a very, very long wait. So how do you reassure patients, during that waiting time, that, while things are cooking, what do you do in that scenario? And the second sort of negative point is, what do you do about people that say, this is for me, but I don't want you to worry other members of the family about this, in that scenario? How do you approach that conversation?
So from our interviews of patients, I think that if you give the patient ahead of time the time frame of when to expect the results back, usually we have a capacity as patients to kind of put it on the side and not to think about it too much, or kind of trying to focus on other things in our life, and as long as we know, as long as we are not close next to the email or the phone waiting for the response. So in the US, I would say now with the companies, takes about six to 8 weeks. So we usually say to patient, it will take 8 weeks, and then usually it's okay. But I think that the people here who had genetic testing can testify as well. Regarding the second question, so first of all, doctors cannot share with family members unless the patient is asking them to. And even if they ask them to, it's usually has to be done through the patient themselves. I think that having conversation with genetic counselors about why it's important to share with family members is warranted. I would really recommend referring to a genetic counselor to discuss that. Family communication can be complicated. Sometimes one of the solutions that genetic counselors have is to write like anonymous letter that patients can send to their family members saying, well, there is a genetic disease that was identified in your family. this is the genetic variant. If you want, you can contact, here is how to contact the genetic counselor to discuss it further without actually providing the name of the individual who was diagnosed, if the individual doesn't want to be like the bearer of bad news in their family. But I think in general, this question of privacy and the confidentiality, it's important to make the difference between the clinical and the research side, and that in the clinical setting, the results are like any other clinical test, they belong to the patient.
That's good. Thank you. There's a question from Professor Matthias Wolf, who's a pediatric nephrologist, and he asks you, how do you think about ADTKD testing in children? So I guess pre-symptomatic testing. How do you deal with that?
So that's a great question. I think that today, the general consensus is not to test children unless there is a benefit for the child. So there are some, the people who say that the children should be tested are usually saying, well, the benefit for the child is that if the parents are less stressed out, it's beneficial for the child. However, they could be also more stressed out if the child is a carrier. Usually the reasoning not to do genetic testing for a child is that the child has the right not to know, the child has the right to decide for themselves when they reach 18. And that's, again, in the case of asymptomatic, which is different from Angela's discussion of her son. Again, I think there is room to have a discussion with the genetic counselors on the pro and cons. And overall, I think in most countries today, the recommendation is to wait for the age of 18. Now, all that could change if there is a treatment and the treatment is showing that early treatment is beneficial, then obviously all the discussion will be different if there is a treatment.
So I hate to interrupt because this is a fascinating discussion and a really, really important one. And so we do have other questions. Unfortunately, we won't have time to get to live, but we will, as I mentioned, we will answer them, post them on the website. One person did just ask quickly if you could share your contact information again.
Sure. I can share it, I think, in the chat with everybody. I'll put it now in the chat.
Okay. Actually, go ahead and share that. And then to the person who asked, we will post all the chat information on the website as well. So if you don't see it in the chat, which I'm not sure attendees can.
Yes, they can.
They can. Okay, great. Thank you so much. I appreciate that. All right. So, and thank you, Dr. Sayer, for helping us understand better sort of the options for nephrologists and to consider all aspects, whether it's the upside downside of genetic testing, but sort of to help nephrologists and patients who are watching today get a sense of the full picture of what the benefits are, what the considerations are, and how it is a very personal decision. So we appreciate both of you for that today. Okay, so we are going to take a brief break. Let me see, what time do we have here? So we're running a little bit behind, but I'm not concerned about that because I think that was a very important conversation that we went a little bit over with. But let me just, before we completely break for 10 minutes, just let you know that we will reconvene in 10 minutes, which is, let's say, 11:40. And please check out our website sometime during the break. Look at these QR codes. And if you're a physician and you want to check out our website on the information we have for physicians, you can see that. If you're interested in genetic testing, we of course do not do genetic testing, but what we do is we collaborate with Dr. Blyer and his team at Wake Forest, where we can get some basic information from you and then refer you on to them for evaluation, for an evaluation or an assessment of your situation. And then, of course, as a nonprofit organization, we're always happy to receive your donation. So we will see you all in 10 minutes. Welcome back, everybody. I hope you enjoyed your break. I got my water so that we can continue here with the rest of our session. So our next session is a conversation around the search for biomarkers of ADTKD, which will be moderated by Dr. Soren Fidelis. Good morning, Soren. Dr. Fidelis is the executive director of the Polycystic Kidney Disease Outcomes Consortium at the Critical Path Institute. Prior to CPAT, Soren was a faculty and entrepreneur at Yale University School of Medicine, where he led a program focused on identifying new targets and pharmacological avenues for the treatment of autosomal dominant polycystic kidney disease. Soren, let me turn it over to you to introduce our first panelist and moderate the Q&A. Thank you.
Thank you so much, Sharon. Good morning, everybody, and thanks for the invitation to moderate this session and to be part of the ADTKD research community. It's been wonderful. So the first speaker today is Dr. Greg Papagregoriou, and I'm sure everybody knows Greg. So he's been in the field for a long time. He's got a long track record in making seminal contributions on ADTKD, in particular the ADTKD MAC1 form. He's currently a group leader at the Center of Excellence in Biobanking and Biomedical Research at the University of Cyprus. He has coordinated the first clinical trial involving CKD patients in Cyprus and has led ADTKD MAC1 biomarker discovery efforts. And previously, he was a research scientist at the Center for Therapeutic Development at the Broad Institute of MIT and Harvard. So I'll turn this over to Greg.
Hi, Soren. Thank you. Thank you for your invitation, Sharon. Well, I do have some slides. I don't see them. Do you want me to share them myself?
I can pull them up, or if you'd like to.
No. I'll do it myself. Yeah, thank you.
Okay.
Okay. Can you see my screen? All right. So I work at the Biobank.cy Center of Excellence at the University of Cyprus, and we've been working for years, especially on ADTKD MAC1. Professor Konstantinos Zelvas was a director, together with Dr. Christoforos Stavru, were the two first people that actually published something on what was called back in the day MCKD1. And this is what we do to discover biomarkers. So we do have lots of patients in a cluster living in the westernmost part of the island, Paphos, and they are more than 160 positive individuals here. And we have tested more than 400 individuals, and actually it's quite impressive that 1 in every 580 people that come from Paphos bears this mutation, the C insertion. So what's the strategy? To develop a biomarker that is, first of all, robust and accessible by everyone so that everyone could participate in trials. It should be multiparametric to include all the peculiar manifestations of the disease, which we're still trying to discover what they are. It should have pathobiological relevance and be disease specific, so that it enables the performance of disease specific clinical trials. So we primarily hold an observational study going on. It's longitudinal, takes years, and basically we do follow and register the natural history of the disease. And then through the biobank, we save data and samples in the form of urine, serum, or plasma, where we go back and test for biomarkers, most frequently for urinary extracellular vesicles, which we're trying to establish as a liquid form of biopsy. Try to see if it actually reflects what happens in the kidney without having to go through a needle biopsy, if that's ever something to achieve. So let's start from the observational study. This is a standard procedure for cohorts that would undergo a trial, especially for a rare disease as ADTKD, diseases as the ADTKD family. Here we have, again, we do have UMOD patients and BREN patients and DNAGB1 patients as well. 11, sorry, but we're focusing on this MAC1 cohort that we study for many years. So we harvest data, we analyze them, we try to build in silico models that simulate the disease progression, try to identify, as many other of the speakers before me said, the reasons why patients have different rates of progression, and then by describing the natural history, understanding the disease, what happens first, what happens second, what happens next, then try to define what will be the clinical trial endpoints. Then this helps us in biomarker establishment because we bring together data from omics analysis and phenotypes together. And of course, this longitudinal type of biobanking enables the possibility to go back and see how something develops in urine or in plasma. Our observational study includes 46 patients, and we have completed 7 years harvesting a lot of data, a big amount of data, and now we entered the 8th year of this study, and we're quite excited. We also included 29 controls, and by controls I mean volunteers that are related to the patients, and they live nearby, so in a way to eliminate the environmental factors affecting the progression of the disease. So we do have lots of data, as I said, and everything, if you see ADTKDs from a distance, everything looks the same. So what we try to do is take a closer look and delineate the phenotype. So we do try to find what type of what defines progression rates. And here you see some words, but how can you justify if someone progresses slower than someone else, and if this person is eligible to be included in the trial? So we do build mathematical models and data sets using machine learning, and we managed to gather data going back more than 40 years from the patients that we curate, 130 patients, and out of these patients we built data sets that were then homogenized in a way by following Euclidean distance protocols, and then we proceeded to do clustering classification of the patients to assist in this search for biomarkers. It's better to have good groups, well-defined groups to compare, rather than having a spectrum that it's not easily being characterized. So this is something that I wanted to show. It's how we define the regions in time that we don't really know what happened, because a patient might check in with their doctor at any point in their lives. So imagine that the beginning of the disease, the first years of adulthood are the data are missing from there. So what actually worked after testing a lot of different versions of this is when we overlaid data that came from the controls that belong to this specific region in time, and also data from the biobank in a machine learning, by using machine learning models, and actually created a very, very robust data set that, as you see here, can distinguish between different groups of progression. And we do use EGFR, we use other parameters as well. Urea is one of them. So urea is a non-specific marker for evaluating kidney function, but here there is a percentage of contribution and it makes every model look better. So we do use it. And here are some results. So you see here that 4 groups in this instance, how EGFR, how the different groups differ from each other as time progresses. And then this is urea as well. And these are other factors like CA15-3, that's MAC1, which we measure in a clinical lab. And then fractional excretion of magnesium has also been an important factor here according to our analysis, magnesium in general in urine, but not uric acid, which it was kind of an important variable for Cypriots once, I guess. So then after having the models coming up with clusters, we try to do some sort of dimension reduction and then tell the algorithm how these patients are classified in order to do forecasting. That forecasting basically means not only to predict the outcome, but to predict how EGFR, how the renal function would be if we had, let's say, weekly or daily or monthly or yearly measurements. The models will go and tell with high accuracy what the EGFR would be like. So you have a trajectory that you predict, and then this could be overlaid to a trajectory being created by through a clinical trial and aims at reducing the time needed for clinical trials in regards to kidney diseases. Then urinary extracellular vesicles. These are small, really small vesicles, like bubbles of the lipid kind of lipid nature that are released in urine by the cells that process the urine, including the cells that are affected in these diseases. So we harvest them from urine, and we do all sorts of analysis. For example, we do multi-omics, proteomics, transcriptomics, metabolomics, and limidomics. And it's a good way to achieve personalized medicine because each patient has their own signature, and it also provides information for new druggable targets, new drug delivery strategies, and also their multi-leveled nature brings multiple dimensions in this biomarker development strategy we're trying to follow, so that it's not just one molecule, but there are multiple molecules interacting. Then there is the biomarker assay development, which is quite important since entering a clinical trial. It would be good to have an assay that specifically measures with great accuracy what the end point of the trial would be if that's a marker from the urine, for example. Here are some heat maps from multiple omics experiments from urinary EVs and also from plasma and urine. And these are the same patients, same time point, same urine sample. And it's really effective to have a biobank because you can move, go back and see how a marker has been developed. Did it increase? Did it decrease? You also have clinical data. You bring everything together, and that's very important to understand and achieve a more disease-specific a way to investigate a disease, and especially, well, this is quite beneficial for rare diseases. We validate the results, try to validate them, come up with a protocol where we isolate rare cells from urine and then do immunofluorescence and see whether protein markers, for example, or pathway markers are expressed in cells that also express markers of tubular epithelial cells. So then it looks something like this. It needs some inspiration and also maybe an artistic kind of view to decide what it's of high importance and not, and then move forward to test it. In the previous years, we did discuss about this biomarker trial that we held some years ago using vitamin D, which was discovered as a molecule that modulates MAC1 wild type and MAC1FS by the Broad Institute. And then we developed an IMRM, that's an immunomolecular reaction monitoring mass spectrometry assay, which we have validated through this trial. And these are some of the results, just to show you that it's quite important to see that each peptide that corresponds to either the wild type or the frame-shifted protein is equally strong, has an equally strong signal, and wild type peptides correlate to each other, while frame-shifted peptides also correlate to each other, but they might not correlate but wild type might not correlate to frame shift, and this is okay. These are two different proteins, and this is how measurement looks look in the Cypriot and the Wake Forest cohorts, and in the graph you can also see CA15-3, which was also an endpoint in this biomarker trial that looks much more dispersed compared to urinary EV signals, and it actually is a good way to establish these vesicles as a means of achieving a real-time clinical trial monitor. So searching for biomarkers in ADTKD, it's a really tough, it's a challenge, but then now there are some opportunities which were not, were only something one could imagine a few years ago. There's a regional interest in ADTKD research, and some speakers before me said about the ADTKNet initiative and the European registry, and we're also trying to, we're part of that, but we are trying to be better, more active. There are observational studies being performed. Tony will also tell you, coming right after me, about their efforts. And we have BRD4780 as the leading therapeutics for ADTKD MAC1 and also for ADTKD UMOD, as published recently by Anna's group. And this brings everything closer towards proceeding to a clinical trial. Hence, we need a really good biomarker, and this is what we are actually trying to achieve. So that's all from me. Thank you.
Thank you so much, Greg. Wonderful talk. Just to double check, Sharon, so we ask questions after Tony's talk, right? Not now, right?
That's right. Let's have
Okay,
Tony
cool.
go next. Thank you.
Perfect. Thank you so much, Greg. All right, so our second speaker needs no introduction, Dr. Anthony Blair. He's our close collaborator right now, and it's been wonderful to work with him the last few months. So Tony, obviously, he's made several contributions in helping identify mutations in UMOD, MAC1, REN, APOE4 as causes of ADTKD. He leads the rare inherited kidney disease research team at Wake Forest School of Medicine, and you know, he's been at it for over 30 years, and you know, we're happy to have him here today. And I'll turn it over to Tony. Thanks, Tony.
Thank you very much, Soren, who's been a great collaborator. We're really excited to be working with him. And I'm just happy to be here today with everybody in the group. It's just been super awesome to see such a great dedication to this area. There are my disclosures. So just really quick, I'm going to talk about biomarkers. Like, we need a biomarker. It's so important, but it has been very difficult to find. I know that, you know, proteinuria is a great biomarker for diabetic nephropathy. And you can basically, test any of these new drugs for a month and see if the urinary protein goes down, and you have something that will likely be successful. And our lack of a biomarker is causing us to have a lot of problems in moving forward with any treatments. So what is a biomarker? A biomarker is a measurable indicator of a biological state. There are several different types. So there are predictive or prognostic ones that predict clinical outcomes. There are diagnostic ones that aid in a diagnosis. There are markers of disease activity. And so here's a good example. This is what we call the Rampoldi score. And so Luca Rampoldi looked at patients with uromodulin, and he did a Western blot where he reduces it, and you can see a lower molecular weight band here corresponding to the uromodulin precursor in the ER that carries endo-H sensitive and glycans. And then there's a higher weight one, and this corresponds to the more formed one. And you can see that we can develop a mutation score based on this. And the more mature, UMOD that we have, the more that gets out of the cell, then that correlates with better outcomes. And so like these patients here with a grade 4, everything is more stuck in the ER and doesn't get out of the cell. And if we look at our grade 4s here on a survival curve of end-stage kidney disease, we see they have worse long-term survivals. In our grade 1 patients, they have the longest survival. And so we can just take now UMOD mutations and look at them in vitro, and we can determine severity. So this is good for de novo mutations, new mutations, especially in young children or young adults where we want to help out. A prognostic now this is good. It's a predictive biomarker, but it doesn't really change, and we cannot really affect it. It's the inherent variant that these patients have. A prognostic marker that's good is, for example, in ADPKD. And here we can look at cyst volume. And this is actually work that Soren had a great influence in, and CPATH really was very instrumental in. And we can see so here's the kidney volume, and this is low kidney volume, and the kidney volume is going up. And when we look at it, we look at the slope loss of GFR, and the bigger the kidney, the more loss of GFR. And so, if we have a treatment like tolvaptan and kidney size decrease or does not increase as quickly, we can look at that as a marker. So that's really another important thing that we'd like to look at. So that's kind of like intermediate, right? Because we can give a medication, and then after a year, if we had some marker of prognosis, we'd be able to see if it changed. But there are also biomarkers of disease activity, and we'd be super interested in this. So an example would be like the sedimentation rate in inflammatory diseases, or proteinuria in glomerular disease, or potentially fractional excretion of urate in ADTKD UMOD. And so we'd like to, and I always say to our patients, it's kind of like your car has a fuel gauge for the gas tank. And the serum creatinine tells us how much fuel we've used up, right? How much kidney function we've lost. But we want really something like a speedometer that tells us how fast we're losing or the activity of disease. And that's something that we would really need. Biomarkers are critical in ADTKD. First of all, the disease is rare, and we cannot perform long-term studies of large cohorts. So when we look at glucose uptake inhibitors, these studies use 3,000 patients over 4 or 5 years. So we don't even have that many patients to look at. Kidney function changes very slowly in a nonlinear manner over time. And I always say that for the patients, the disease progresses rapidly because they're seeing family members go on dialysis and or needing a transplant, and they're seeing themselves getting closer to transplant. For clinicians, it goes relatively slowly, We see the creatinine stay stable for a year or two, then we'll have a little rise in the creatinine, and it just kind of slowly goes up over time. But for researchers, the progression of disease is agonizingly slow. We have a change in GFR of 2 ml per minute per year, and it just makes it so difficult to study that we cannot really follow eGFR as a long-term outcome in a study. Daily fluctuations in GFR are also a problem. So you're a little more hydrated or less hydrated. And then non-GFR fluctuations in serum creatinine, like if you had a steak or a cheeseburger the night before, you get your creatinine, that can cause variation. And so these are also problematic. So the biomarker depends on the clinical trial that we're going to do. So say we did a trial of a drug that just overall decreased UMOD production, then we would be able to look at urine or plasma UMOD. And a good thing is both of these things are easily measured. We could say have a drug that just decreases mutant UMOD production. So then we will look at urinary mutant UMOD, and this is harder to do and is still subject of investigation. We could also, as one of our speakers mentioned, like the common final pathway is tubulointerstitial fibrosis. So what if we just had a drug that lowers tubulointerstitial fibrosis? And so that would be one of these biomarkers that I'm always perusing journals and trying to find, or biopsy, if we could look at a biopsy of patients before or after, or imaging. So similarly with MUC1, now MUC1 is made throughout the body. UMOD is cleaved by hepcidin and goes into the urine. MUC1 is only in the exosomes or within the cell. It's not cleaved. And I think Greg is looking at these exosomes and doing an awesome job. So again, we could look at, if we overall decrease MUC1 production, we could look at urine or plasma MUC1. But we have to know that it's also made in the kidney and the bladder. So it's what are we studying when we give a drug? Is it decreasing production in both the bladder and the kidney, and how does that affect things? We could look at decreased mutant MUC1 production. This would be difficult if we looked at just the kidney, but if it decreased it all over the body, we could do, for example, biopsies of the salivary glands or the sebaceous glands. Slowing of tubulointerstitial fibrosis, again, biomarker biopsy or imaging. Mutant MUC1, so what are advantages and disadvantages of biomarkers? Mutant MUC1 is present in exosomes. MUC1 is made in bladder and the kidney. Mutant MUC1 is difficult to measure. So this is a tough one. Previously studied biomarkers. So there are a lot of biomarkers that have been studied over time. But when you look at a lot of the papers for these biomarkers, they usually say, this biomarker looks good and it predicts outcome. However, after we adjusted for urinary protein and baseline kidney function, it was no longer significant. We've tested a lot of biomarkers. We're interested in testing more. Imaging. Images is an exciting area. The big problem with this is we can't go back 5 years. We don't have imaging from 5 years ago that's specific, looking at specific detailed mechanisms of imaging. So any imaging studies that we do are going to be difficult, and we're going to have to just do them looking forward. And again, there are different areas of the kidney that we have to look at. The kidney is shrinking over time with this disease. And so we think these are problematic, though promising. Biopsy is also something that could be really helpful. However, there's sampling error with a kidney biopsy, right? We get 20 out of 1 million glomeruli. There has not really been a good cohort biopsy study. We are actually pursuing this right now. We are doing, so if anyone has kidney biopsies on a number of patients that they'd like to scan and work with us on, this would be really great. And if you could contact us, we'd like to build a big library together. Optimum study is a study that we're working with Soren on right now. It started September 2024. Our goal is to accelerate discovery of potential biomarkers. And the good thing is, first of all, we've collected a lot of data since 1996, and now we're going to get a lot of statistical analysis. We're going to follow patients prospectively for 3 years, and then we're also going to look at ultrasound and elastography of the kidney. And so basically, we're going to look at kidney stiffness and follow this over time. So biomarkers are specific biomarkers that we see throughout the kidney, and we would think, for example, biomarkers in the thick ascending limb or distal convoluted tubule would be helpful. Here are the biomarkers that we've looked at Wake Forest over time, and unfortunately, we haven't found any that we think are really primed to look at for kidney for a study. We've looked at KIM1, NGAL, TNF, DKK3. You can see just a wide variety of these. So we have a baseline data set of 150 patients, really well phenotyped with lots and lots of baseline urine and plasma. So if anyone has an idea for a biomarker and they're like, wow, we'd like to test your samples, we're so happy to share this with anyone. We looked at KIM1, and here are our patients with KIM1, just looking at their EGFR decline cross-sectionally over time. And we didn't really see anything with KIM1. We were been hopeful that that would work. We looked at MCP1. Again, we did not see anything significant over time with that. So we're really excited about working with Greg, because Greg is just looking at everything through a more like an ohm analysis without any bias. And we'd like to take anything that he develops and hopefully validate it in our cohort, or if anyone else has biomarker ideas, we'd be super interested in working with you on this. Our ALEC, we can look at genetics, demographics, plasma, and urine. So we're very interested to share samples, and here are our many collaborators. But sad to say, right now, we still do not have a really good biomarker of disease activity. It's really kind of hampering our work. And if anyone has any good ideas, we'd love to work with you on this with our samples. So thank you very much.
Thank you so much, Tony.
Yeah.
Wonderful
Let's
talk.
see here. Let's see, am I still sharing?
Yes.
All right, screen sharing. Let's see here. Let's see if I can get out of this. Some reason it won't let me get it out of it. Can you guys just stop the screen sharing? Oh, here we go.
Yeah.
I got it. Stop share.
Yeah.
There we go. Okay, sorry about that.
Perfect. So we have a couple of questions. Actually, Sharon, do you want me to ask the questions in the chat as well?
That's fine. If in the Q&A, sure.
Sure. Perfect. So the first one is for Greg first. Have you considered using machine learning algorithms like LSTM, which is a type of neural network that can be applied to higher dimensional data to improve prediction accuracy?
Yeah. I didn't have enough time to show you more stuff, but we are trying to build some sort of a digital twin of MAC1 affected tubules, how they behave, because we don't have all this data now, and there are also data from the literature and also data in freely available like databases and everything. So if you bring everything together and you have weighted contributions, then you can ask questions, and the twin would make decisions for you, and it will tell you which direction to look at. And that's one of the solutions that we are trying to explore. And in general, like neural networks is something that might be more effective to use here than other types of interconnecting systems. So yes, that's a really good question. And hopefully it will pan out at some point. It's very, very hard to achieve.
Thank you, Greg. And this is more general, I guess, because Tony alluded to the difficulty of finding a biomarker of disease activity. Can you, for both of you guys, can you comment why it's been so hard to do that? And, secondly, if you had one available, what would be the first study that you would do? And let's start with Tony.
Yeah, it's a good question. We have a genetic disease, and so unlike other conditions, we can start from young patients and get samples. I think it's just because the disease there's no specific inflammation or any type of specific dynamic that leads to inflammation that we could measure. The first study I'd like to do is well, there are several. For the patients, I know they always ask me this. I'd love to do a vegetarian diet and see what that does, or a Mediterranean diet. And then I'd like to look at losartan or other ARBs to see. And that would be just so great if we could just start with basics like that.
Thank you, Tony. And Greg, any comments?
Well, just I just want to say that it's a really important part of what we are trying to succeed here, to find robust biomarkers that go beyond diagnosis, but also can be used in trials as well. Some things that change. So if we have access to samples, access to data, then that makes our life much easier. And we're very thankful to our patients here in Cyprus. They are very responsive and very involved. And we are very keen in keeping this collaboration that we have, because it's really, really hard to identify something that makes sense and that works in the hands of everyone. So just a big thank you going out to patients for their support.
Yes, if I could just say something too, I should have mentioned that first. Yeah, the patients are just so important. And so every data point is a patient, right? And every point brings us closer to where we need to go. And when you have a rare disease, like Greg is saying, every single patient just helps us with this.
Yeah, especially now that we're living in the era of personalized medicine, which means that each patient can basically act of their own control, and the patient itself can provide the information of what is needed to be provided as a therapeutic intervention, or how things are measured. I mean, everyone is quite special to have their own mechanistical aspects that signify how the disease behaves. So that's quite important. And a rare disease brings us along this type of data sets that are patient specific, and that's really important for us, but also hard to manage.
Thank you, Greg. And yeah, I want to say, wonderful talks and, sessions. And this is really, how the PKD field started, right? 25 years ago, people were kind of doing their own thing, collecting data. And I think it's important that people are talking to each other, creating this databases that can be leveraged, ideally moving forward to federated analysis where we can look across different registries and databases for different, biomarkers, disease patterns, et cetera. And this is how, a field matures and you get to a prognostic biomarker or a drug response biomarker, and that's where the pieces come together the way they have for ADPKD, where TKV now can be used as a primary surrogate endpoint, or reasonably likely surrogate endpoint. So yeah, very grateful to be part of this community.
Thank you so much, Soren. We're going to have to move on, but thank you, Soren and Tony and Greg, for this really important and forward-looking presentation. So we're very excited. Thank you.
Thank you.
Okay. Our next presenter is Dr. Silvana Bazua-Valente. Dr. Bazua-Valente has long been captivated by the kidney's structure, architecture, and specialized cellular processes, a fascination that first took shape during doctoral studies at the National Institute of Medical Sciences and Nutrition, Salvador Zubrian in Mexico. Today, as a research scientist at the Kidney Disease Initiative led by Dr. Anna Greca at the Broad Institute, she now strives to translate those discoveries into tangible therapeutic strategies. Thank you for joining us, Silvana.
You can you hear me?
Yes, we can, and we can see your screen.
Excellent. Okay. And can you see? Okay.
Your slides are small because we can see your notes pages. Would you rather I share the slides and you can look at your, oh, there you go.
Perfect. Okay. Good afternoon. Thank you for the opportunity to present my work here to all of you in this very important symposium. I am very honored to share our work on a potential new strategy for ADTKD caused by UMOD mutations. So without further ado, let me start by sorry. Okay. Let me start by starting to talk a little bit about the disease. ADTKD UMOD is a rare inherited form of chronic kidney disease driven by mutations in the UMOD gene. It causes progressive kidney failure, often leading to end-stage renal disease relatively early in life. And with over 135 mutations, and due to its rarity, ADTKD UMOD remains significantly underdiagnosed, highlighting the importance of increased awareness and genetic testing for patients with familial kidney disease. Currently, as we have heard from all the previous speakers, there are no treatments that target the underlying cause, and this lack of specific therapies creates a significant burden for patients and really underscores the urgent need for new strategies that stop disease progression. And this is why our goal is to uncover the fundamental mechanisms of this disease to identify novel therapeutic targets. So the key protein in this story, the star of the movie, is uromodulin, or UMOD. UMOD is produced exclusively in the kidney within the cells of the thick ascending limb of the loop of Henle. UMOD is a GPI anchor protein, meaning that it belongs to a family of proteins that use a very specific glycolipid anchor to reach the cell membrane. Normally, UMOD is synthesized and folds in the ER. It travels through the Golgi, where it's glycosylated and the GPI anchor is added. And then this anchor directs it to the apical membrane. And this apical surface, do you see me pointing on the screen? Okay. So this, once it gets to the surface, it faces the lumen of the tubule, which means that once UMOD is here, it can be readily secreted to the urine. And indeed, as we've also seen from our previous speakers, it is the most abundant protein in normal human urine. Notably, when we began this research 5 years ago, the specific trafficking partners responsible for guiding UMOD's journey were unknown. UMOD has several structural domains that are critical for its function. 95% of mutations cluster in exons 3 and 4, which encode the EGF-like domain, EGF-like domains and the cysteine-rich DHC domain. About 60% of the mutations directly affect cysteine residues, disrupting the disulfide bonds that are essential for the proper folding of the protein. And this misfolding prevents UMOD from exiting the ER, so it gets it trapped intracellularly in the ER, eventually triggering ER stress. This electron micrograph, adapted from a review that was written by Olivier Devoise, who's an expert in the field, shows the consequence of the consequent ER stress. Compare the very massive and hypertrophied ER cisternae on the top panel versus of a tubular cell of an ADTKD mouse model compared to a normal ER structure of a normal healthy tubular cell on the bottom. The chronic ER stress eventually damages the tubular cells, and this damage triggers downstream inflammation and fibrosis. So there is active recruitment of inflammatory cells, there's activation of the fibroblasts, which then they secrete extracellular matrix deposition, and so this scars the kidney interstitium and ultimately causes the kidney failure. Previous therapeutic strategies in ADTKD mouse models have often focused on mitigating the downstream consequences. For example, they have focused on targeting inflammation. But for this work, instead of looking downstream, what we aim to do is to understand the root cause. So we aim to understand and ask the question, who is interacting with misfolded UMOD? So building on prior work from other labs and also our own lab, implicating a family of cargo receptors called the TMED cargo receptors. These cargo receptors have been implicated in trafficking of misfolded proteins. So we hypothesize that TMEDs might also be involved in the intracellular trafficking of UMOD. So to investigate this directly, what we did was we utilized an ADTKD mouse model that carries a mutation in a cysteine 125. This is a mutation of a cysteine to an arginine. The mutation is located in EGF-like domain 2. We then performed immunofluorescence staining on kidney sections using antibodies against UMOD, which you can see in red, and the cargo receptor TMED2, which you can see in green. And as expected, in a healthy wild-type mice, UMOD correctly localizes in the apical membrane, which you can see facing the lumen of the tubule. And in contrast, in our C125R mice, the mutant is retained intracellularly. And you can observe a stronger co-localization indicated by the yellow signal between the trapped mutant UMOD and TMED2. This provided the first direct in vivo evidence of TMEDs being part of the UMOD entrapment. Now, let me take a step back and tell you a little bit more about the story of TMEDs in our lab. This came by foundational work by my friend and colleague, Moran de Vela-Levitt, who's shown here in this picture with our mentor, Anna Greca. Well, in the Greca lab, Moran was studying ADTKD caused by mutations in MOG1. And similar to UMOD, mutant MOG1, which you can see on the top panel in green, is retained intracellularly. Then Moran made several discoveries. First, she found that MOG1 was trapped in vesicles containing TMED9 cargo receptor. Then, through meticulous screening, she identified BRD4780, which is a compound that could efficiently clear the trapped MOG1. And you can appreciate this on the bottom panel, where the mutant protein is completely cleared. And mechanistically, she determined that BRD4780 works by binding to TMED9 and thus releasing mutant MOG1. So this groundbreaking work was pivotal, and I hypothesized that it could be also a strategy that we could apply to ADTKD UMOD. So we tested this in our ADTKD mouse model. We treated mice daily with either BRD4780 or vehicle for a month, and then we performed single tubule isolation to specifically examine UMOD localization, and the results were strikingly clear. This is a longitudinal view of the tubule, so to help orient you, I added the arrow which is pointing to the lumen. So in the vehicle-treated mice, which is the top panel, you can see mutant UMOD accumulated intracellularly with TMED9. But in mice treated with the BRD, which is the bottom panel, there's a clear reduction of TMED9 9, and there's also a very clear reduction of the accumulated UMOD. And very importantly, we could see how UMOD localization is restored in the apical membrane, so it's facing the lumen. So this was very exciting. But then we asked, how is BRD 4780 eliminating the toxic accumulation? We hypothesized that it could be by 2 ways. One is that we're promoting the secretion of the protein by reestablishing its normal trafficking to the apical membrane, or the other is that we were promoting lysosomal degradation. So to solve this, we turned to a cell model where we expressed mutant UMOD. We found that BRD promotes the movement of mutant UMOD out of its trapped state in the ER quantitatively, as you can see in the graph on the right-hand side. We analyzed the change in colocalization compared to the control cells. And you can see how there's a significant decrease of the association of UMOD with early secretory pathway markers. And conversely, there's an increase in colocalization with markers of the later pathways. Specifically, we observed a clear shift towards the late endosomal and lysosomal pathway, which is marked by 2 proteins, EEA1 and RAB7. Now, in the images that I'm showing you, we utilized a very powerful cyclic imaging technology called 4i that was pioneered here at the lab by my colleagues Jason Sabras and Matthew Brown. And this allowed us to see the same cell, but multiple times with different antibodies. So if we see first the control cells, which is the top panel, you can see UMOD in red, heavily clustered near the nucleus with a very high overlap with TIMET2, which you can see by the yellow signal, and very minimal or almost nothing of overlap with EEA1 and RAB7. In contrast, after the treatment, UMOD, first of all, is sparse throughout the cell, and you now see more points of colocalization marked by the yellow spots with EEA1 and RAB7. So this strongly suggested that BRD is facilitating the release of UMOD to the endolysosomal system and likely towards degradation. But biological systems are rarely simple, and it is never one thing or the other. So we also wanted to see if we were also getting more secretion in the urine. And this can kind of relate to what Dr. Blair was saying about the biomarkers. So what we did was we collected urine from the mice before and after the treatment. And then we isolated UMOD from the urine, and we performed Western blot analysis with an antibody that recognizes only the mutant UMOD. And very remarkably, as you can see, from the panels that are highlighted by the red background, the BRD 4780 significantly increased levels of mutant UMOD excreted in the urine of the mice after the treatment. So compare the columns of the endpoint versus the baseline, but also overall, you can see that that is very distinct how the BRD increases significantly the mutant versus mice that were treated only with the control. So to summarize up to now, BRD helps clear mutant UMOD, but our next question was, does solving this intracellular problem actually mitigate the downstream kidney damage? And to answer this, we assessed inflammation and fibrosis in our mouse model. We stained sections for CD45, which is an inflammatory cell marker, which you can see in purple, and alpha-SMA, which is a marker of fibrosis, which you can see marked in brown, in a brown reddish color. The treatment, excitingly, after one month of treatment, significantly reduced both inflammation and fibrosis. And when you compare the histological panels, you can see how with the BRD treatment, it actually, these parameters are restored very similar to what it looks in a normal, healthy, wild-type mouse. Finally, to assess the relevance in a human disease, in the human disease, we generated kidney organoids using cells derived from patients carrying 2 distinct UMOD mutations. So compared to the organoids from the healthy control, which you can see on the left-hand side, where UMOD is in red, and then we have MOC1 as an apical marker, you can see how UMOD is correctly localized in the apical membrane. But the patient-derived organoids faithfully recapitulate what the disease phenotype, and they show more mutant UMOD trapped intracellular, so almost no localization in the membrane. When we treated these organoids with BRD, we rescued UMOD trafficking, restoring its localization very clearly in the apical membrane, and which is exactly what we observed also in our cell models and in our animal models. So to briefly summarize the key findings, first, we established that the intracellular trapping of UMOD is mediated by TMED cargo receptors. Second, we demonstrated that pharmacologically targeting these TMEDs releases this entrapment. Third, we elucidated that the mechanism of rescue is a dual action. On the one hand, it enhances the secretion of mutant UMOD in the cell, but also it promotes clearance via lysosomal degradation. And fourth, we also found that this strategy proved effective to reduce inflammation and fibrosis in our in vivo model. So to conclude, what is the broader impact of this work as we look ahead? Well, our findings established TMED cargo receptor as a novel druggable target, offering a strategy that addresses the root cause of ADTKD UMOD by correcting the fundamental protein trafficking defect, rather than managing the downstream consequences. And then I believe that the most rewarding feeling comes from seeing a discovery go to an actual cure. So to effectively build upon this discovery and move towards a therapeutic reality, our key next steps involve developing preclinical tools that will allow us to do a better testing and a better testing of the therapeutic responses. So right now we're particularly focused on creating innovative cell models and biomarker tools, as our previous speakers were mentioning, to all of these obtained non-invasively from the patient's urine. And because the ultimate goal is to successfully bridge the gap between the promising preclinical findings and effective clinical applications for rare kidney disease. And this might not be only BRD, but it may be all other potential strategies that come in the future. So we will keep on working to build a strong foundation for the future of therapeutic development in ADTKD UMOD. Finally, I just want to thank, I really want to say first that science is never a one-man job. And so I have a lot of people to thank. I do first want to thank all the patients, because the patients are a great inspiration for what we do. It's very inspiring how the patients fight against this disease very strongly and very gallantly. I really want them to know that whenever they're feeling weary, we are back on their backs, and we will keep the torch of the hope on. I also want to thank all the GRECA lab members, of course, some of them that indirectly or directly were part of this work, our collaborators all around the world, and I, lastly, but certainly not because of the last, but I do want to thank the person who really made this possible, which is Dr. Ana Greca. She is the one who brings all of this together, the patients, the researchers, the people, the funding, of course, but really everything is thanks to her. I would certainly not be standing here in front of you if it wasn't for her. So I really want to thank Anna, who's a real force of nature. Thank you.
Thank you so much, Silvana. Very good props, due props to Anna, who's coming up next. And I just have to say about your presentation, I think that is, for as technical as it was the most inspirational technical presentation I think I've seen. And so thank you for you and your team and all the work you're doing on these breakthroughs.
Thank you.
Okay, let's see, Kathy, do we have questions for Silvana?
No, let's start with David's question. He states, this is a powerful potential therapeutic. With powerful therapeutics, there are risks of greater side effects. Have you observed any side effects in treated mice?
Yes, the side effects we have seen in mice. However, we have managed those side effects by controlling the dosage. And again, I wouldn't necessarily think that this would be something of a future problem, because it also depends on the metabolism of the study system. And I think that maybe it could be also because of the mice and not necessarily because it's a side effect of the drug.
Thank you.
So that's good to know that you're monitoring side effects even at the developmental stage and long before it gets to a clinical trial.
Yeah, of course, of course.
We have another question from Anne. Are these findings transferable to MUC1?
Yes, they are. All of this work on ADTKD UMOD was done because of the foundational work that was firstly done on MUC1. So yes.
Okay. Another question concerning in vivo studies with BRD 4780. You treated about 10-month-old male mice. The first question, there's two questions. Did you also treat younger mice? And does the compound need a certain ER accumulation of misfolded UMOD before a noticeable reuse of the phenotype can be detected?
That's a very good question. So I think I'll answer in two parts. The first one was if I treated mice younger.
Yes.
We did not treat mice that were younger than 10 months. The reason is because to assess the outcome, we wanted to have a window where we could really see a difference between the genotypes and where we could see really a phenotype of the disease. So we treated older mice. However, I do want to point out, and that's kind of answering the second question, we do know that BRD 4780 works also on the wild type UMOD by enhancing its secretion. So it's not only the effect on the mutant protein, but it's also the effect on the wild type, which is actually pretty good, because then we know that this strategy can be targeted in various ways and not only for the mutant protein.
Well, thank you. And then we have one more question. Does the mutant MUC1 UMOD protein function properly after reaching the cell surface upon treatment with BRD 4780?
Yes, that's exactly what we think and we're excited about, because we are not only getting rid of the toxic accumulation of the mutant protein, but we're making the wild type protein get back to where it has to be and do its proper function to reestablish all the kidney function in a normal way.
Thank you very much. That's all the questions we have.
Thank you so much, Silvana, for joining us. Fascinating and just so exciting to think where this research could lead. Thank you. Okay, next up, we're running a little behind, but I hope you'll stick with us for the entire rest of our final session here. I'd like to introduce Richard Nelson, co-founder and the board chair of the Rare Kidney Disease Foundation, who will moderate this final session of the day. And as a quick introduction of Rich, after generations of family members who died quite young from kidney failure, Rich obtained a transplant 34 years ago and only recently discovered, though, the cause of his family's disease. And he, like I and many others, have MUC1. He and his nephew established the Rare Kidney Disease Foundation in 2018 to raise awareness of this disease, to provide support to patients seeking answers, and to support physician scientists like Dr. Greca and her team and Dr. Tony Blyer and his team in their quest to find a treatment and ultimately a cure. So, Rich, it's all yours.
Thank you, Sharon. And Sharon doing a great job. I mean, what an executive director. You're doing a super job, Sharon. Thank you very much for bringing us all together. I've been eager to hear what Dr. Anna Greca is going to present today about therapies on the horizon, especially since these 2 days are all about the time is now uniting to halt ADTKD. Dr. Greca is a physician scientist, a professor at Harvard Medical School and Mass General Brigham, and on the executive leadership team at the Broad Institute of MIT and Harvard. I hope each of you have seen the TED Talk. It was a phenomenal presentation that was given 2 years ago. This wasn't just a normal TED Talk. This was a phenomenal TED Talk at the highest level of the TED organization. It's all about the world's rarest diseases and how they impact everyone. It was inspiring. It was very hopeful. And we're all eager to hear today a update on the future. After being encouraged 8 years ago by Dr. Greca, we started the Rare Kidney Disease Foundation. As Sharon has described, it's all about being patient-focused. It's all about being patient-driven. And it's an organization run by all volunteers. All of us either have MUC1 or UMOD or related closely. It's all about, accelerating, finding, I mean, in our world as patients, timing is everything. I mean, it's life and death, and it's a lot of suffering for a lot of people. So the holy grail that you as scientists and you as physicians and you as researchers is everything to us, and anything we can do to support and help get a drug-like compound to the FDA, we're all in. And that's why we exist as a foundation. I've also found Anna as profoundly motivated and committed to patients more than any other physician, scientists, and organizations that I've been with over the last 4 decades. As brilliantly presented in her TED Talk, it's all about sleuthing molecular science to bless us as patients. And it's not just a few of us anymore. It's literally hundreds of thousands of families are affected by ADTKD. So let's turn it over to Anna. Anna?
Richard, thank you. This is a very generous introduction, and I'm deeply honored to be here. I also want to offer my sincere congratulations to Sharon and everyone else at the Rare Kidney Disease Foundation. It's just remarkable how much the organization has grown, and I'm just so delighted that we're raising awareness, and the caliber of the speakers today, I think, speaks for itself. Also, the future for the field is bright. with all my young colleagues who have spoken today. So it's just so wonderful to see, patients and scientists and physicians, all the stakeholders really coming together. So very heartwarming, and I'm very honored to be a part of all of this. So really terrific, and congratulations to everyone. So I guess I'll share my screen and give you very briefly an update. I know that it's late in the day and folks are probably tired. So I hope you can see the proper window for the title page for the presentation. So the goal is to really give you a quick update on therapies on the horizon. These are my disclosures, and they're relevant to this work because, we have been building a company that licensed some of our discoveries out of the Broad and was funded by Atlas Venture and Omega Funds, and is now partnered with Alexion and AstraZeneca on its way to a hopeful outcome to have something that we can take to the clinic. So that's a relevant disclosure for my work today. I only act as an advisor to this entity, but nevertheless, I care deeply about the stewardship of this discovery all the way to the clinic, and I do whatever I can to make sure that it makes it there. So I just wanted to take a step back. Silvana obviously beautifully and eloquently represented our team and our most recent discovery. But just to take a step back, the work that we do in the lab is really starting with human genetics, like ADTKD, MUC1, or UMOD. And then our sort of specialty, if you will, is that we use what I call curiosity-driven science, so classical scientific inquiry, partnered together with scalable technologies, things that allow us to monitor multiple things at the same time in human cells or in human systems, like the organoids that you just saw from Silvana. And so we study cellular mechanisms because it's not enough to know the gene. What you need to know is the mechanism in which the gene is involved to essentially perturb the circuits inside the cell. And then, of course, once we understand what that perturbation is, then we can try to fix it. So this is, the hopeful outcome of our work, that we can come up with medicines, candidate medicines that we can take forward toward the clinic. And so over the years, how have we done in terms of this? So in work that we are not going to be discussing today, and it's not relevant to ADTKD, but is relevant to work that came out of our lab, is just to show you that we have done this before. And so early on in my work, starting out my lab, we worked on a different kind of set of mutations that involve an ion channel that ultimately is important in a pathway that leads to another kidney disease that is sometimes, as a diagnosis, also implicated in the constellation of findings that one sees on a biopsy, as some of our colleagues mentioned earlier, but the disease is called FSGS. This is a genetically defined form of FSGS that we were studying. And I just wanted to show you that the discoveries out of our lab made it all the way to phase 2 trials in patients, where a number of patients had really good response to this drug. And it is now partnered with Bristol Myers Squibb on its way to hopefully follow on trials in the future. But just to give you an idea as well, that discovery was made several years ago, as you can see from our publications, and it's still not done. And just to show you that the humbling thing about us as physician scientists is that, drug development based on our discoveries takes a very long time, and there are many hurdles along the way. So it's very hopeful and wonderful, and we know how to do this work, but there are many unexpected twists and turns along the way. And so I just want you all to come along with us on this journey with the understanding that it can take quite a long time, although we're working as hard as we possibly can. So now this brings us to the work that we want to talk about today, which is, as you heard all morning about MUC1 mutations and UMOD mutations. Several years ago, when we initially published our work on MUC1, everybody in this field was asking me, what about UMOD? And I kept saying, it's coming, we're doing the work. Well, now you saw how hard Silvana has been working over the last several years to get all of this work done for UMOD as well. But as you can now appreciate, we have rounded out our findings both in vitro and in vivo, meaning in cells and in animals, that really show us a road toward hopefully developing a TMED-targeted compound that can help with ADTKD, with phase 1 trials planned for sometime in 2027 or beyond, depending on how lucky we get with the drug development process from this point forward. So again, there are possibly unexpected twists and turns, but we're on our way. And so as just a summary of the MUC1, there was a question earlier. This is prior work that was published several years ago, but we did have all the same evidence that Silvana showed you for UMOD. We had initially gathered it for MUC1. This is work that was done by a team at the Broad, led by Mohan Vela-Levitt, who is now a leader of her own lab at the Bar-Ilan University. And it was just really wonderful to see that we had a tool in this BRD 4780, not a drug, but a tool that could help us understand the mechanism of the disease and then also light a path to a possible therapy. And as you just saw from Silvana, this is just a little sampling of some of her work to say that this tool compound is again working for UMOD. This is the mutant UMOD that the compound results in the degradation of. And so again, hope that we have something that illuminates a path toward a therapy. This in itself is not a therapy. This is a tool compound. But based on this, we are now sort of on our way to develop therapies for ADTKD. I do want to let you all know, as I have been saying, that our first effort to develop a compound that was inspired by 4780 ended up with an unexpected toxicity, just as we were about to file an IND, which is, you know, what you need to do in order to take something into patients. So there was a late stage toxicity that was uncovered. For those who are experts in this space, I will say that it was an off-target toxicity. So it doesn't have to do with the mechanism of the drug on the TMEDs, but it had to do with something unrelated to that we identified. And so that had to stop. It wasn't safe to move forward with patients. So this is exactly what I mean when I say twists and turns in drug development. And so we're trying again. And this is an ongoing process. There's a dedicated team of scientists who are working very hard to make this happen. And we're hopeful that they will be successful in building a new drug that can then make its way to clinical trials. I'm very hopeful that this will be solved and that we will have positive news moving forward. But we're very much in that process of trying to work this out. So just so that everyone understands that, you know, drug development is not linear and there are twists and turns along the way. Now, something else that is very interesting about the discoveries that we made, while today we're focusing on our discoveries in the kidney appropriately because they're focused on ADTKD, it is actually very interesting and powerful that this TMED targeting mechanism is actually not only relevant to diseases that involve misfolded proteins in the kidney, like UMOD and MUC1, but this TMED system is actually important for additional misfolded proteins that cause other diseases in other families. So they cause a familial kind of Alzheimer's disease, a form of Alzheimer's that runs in families, a form of blindness that runs in families called retinitis pigmentosa, and additional diseases in the liver, in the lung, other places, which we are currently exploring. And this is important because this is a nodal biological mechanism, is what we call it. And the reason that it's important is not only can we help more patients potentially by doing this research, but also it is more advantageous for the development of a drug for these diseases, because you can obviously increase the number of patients that a pharmaceutical company can target by taking this therapy forward. So this is something that's, I think, of wider interest. And so, again, sort of coming back to the initial brief description of how our work proceeds in the lab, this is a story in which we started with these very ill-defined and poorly understood mutations. We understood the mechanism through which these mutations work to break circuits inside cells, using both our scientific curiosity as well as our scalable technologies. And we now have sort of a candidate strategy for what we can do take forward in the clinic that can not only help patients with UMOD and MUC1 mutations, but potentially many more patients down the road. So very hopeful that this work will actually catalyze progress for a lot of patients. And just as my final slide, I would like to say that while this small molecule shown here is our first climb toward the peak, with the ultimate goal of preventing, curing, or completely managing the disease for all patients. This is only one of our efforts. In the lab, we also have additional efforts because one can not always rely on the small molecule, the pill being the answer, which is sort of what we have been talking about today. We're looking at next generation treatments, which is ongoing work in the lab, looking at things like antisense oligonucleotides and siRNAs that can be targeted to these diseases. And of course, we're also interested in what someone would call a one and done therapy, which would be to insert a gene therapy, with scissor-like or tweezer-like precision, a way with which to fix the gene inside the kidney cells of patients so that they will never have to worry about the disease. Especially if we intervene very early on in young patients, we would be able to essentially, have a one and done approach of curing the disease once and for all. So these kinds of strategies are far into the future. I want to make sure you hear me say this. We have potentially a decade ahead of us before the gene therapy can become a reality for the kidney disease. But we are obviously starting now. The climb, as you can see, is going to be long and hard with a lot of twists and turns. But we're up for the challenge. I'm blessed with a tremendous team of collaborators, and we're really eager to do this. As a final word, you may have heard that we have recently founded a new entity, a new platform at the Broad called the Ladders to Cures Accelerator, whose mission is really to catalyze progress across the research ecosystem for all patients with rare genetic diseases, not just in the kidney, but across the board. There's great unmet need. These rare diseases affect, 30 million patients across the United States and many more around the world. More than half of those patients are children. So we really want to make progress based on what we have learned from all of this work to benefit more patients. And again, the strength of the team is each individual member, and the strength of each member is the team. I firmly believe this. I think that this is really embodied by this wonderful group of collaborators that I have the great honor and privilege to work with every day, as shown here. This is our last lab retreat. Most grateful to all of you, my collaborators across the world who have been here today, and also patients, of course. This partnership with the Rare Kidney Disease Foundation and patients everywhere has been incredibly powerful. And again, I'm so happy and proud, if I can say, to see all this wonderful progress that has been made. And of course, we have a long road ahead, but together, I'm confident that we can tackle all these diseases and make good progress. So thank you for inviting me today, and I'm happy to answer any questions if we have a little time.
Thank you, Dr. Greca. We do have one question. For patients who are located outside of the United States, how do they become part of clinical trials when they become available?
Yeah, I think this is clearly, as I mentioned to you before, when we license the technology out to a startup company and then in partnership with a pharmaceutical company, we are grateful for these colleagues who are making an investment into bringing this to clinical trials. As you can imagine, it takes literally millions and millions of dollars to get a therapeutic strategy all the way to the clinic. And so those decisions about how the clinical trials will be deployed will be made by our colleagues. Of course, we work together as a team. We advise our colleagues as best we can. And in my mind, this disease is rare enough that we may not be able to enroll all the patients in the United States. And in fact, there have been conversations about enrolling patients outside of the United States in other countries. So obviously, our colleagues in Cyprus have a wonderful and very well organized community of patients there. So, it would be an obvious place for a clinical trial to go. But of course, there's many other places in Europe. There's actually patients in Asia, in Australia. There's patients everywhere on the planet who are affected by UMOD and MUC1 kidney disease. And so, again, it would not be up to me, but I'm hopeful that our partners will deploy clinical trials in other countries as well. I think if I would, make any additional comments on this topic, I would say that what the Rare Kidney Disease Foundation can do and what each and every one of the patients can do is to actually become involved, become part of the registry, because only if we can count you can we then know about you or other members of your family who might be willing to participate in clinical trials. The obvious advantage is that that gives you early access to treatment. And of course, by the time it makes it to a clinical trial in patients, this treatment will have been determined to be safe. And so it will be looking for efficacy, what we call phase 2 trials. So, please have yourself counted, have your family counted by participating in a registry, raising your hand to participate in the Rare Kidney Disease Foundation. I think this is the best way for us to know what numbers of patients are where, in which locations, so that we can deploy clinical trials in those locations and hopefully make the treatment as widely available as possible when the time comes. So I thank you for the question. It's very important.
Thank you, Dr. Greco. That's all the questions we have right now.
Perfect.
Okay. Thank you, Dr. Greco. In the decade that I've known and worked closely with Dr. Greco, she really has taught me about, she is the ultimate collaborator. That's why the patient advocacy organization works. That's why we're having this discussion today. We essentially, over the 2 days, we'll have around 200 participate. We invite you tomorrow to the patient day, but many thanks, Anna, for your brilliance and your team and allowing us to contribute too.
Thank you
Back
for
to
having
you,
us.
Sharon.
Thanks, Rich. And let me just quickly close here. I know we're just a couple minutes over. We did pretty well. And I think it helped that Anna spoke at about 100 miles an hour. But thank you so much for packing so much in a short period of time. And if we do get additional questions for Dr. Greco, again, remember, we will answer them and post them on the website. So don't stop asking questions if we didn't get to your question. Wrapping up, I quickly want to share this QR code, please. Before you go, take a picture of that and fill out this 3-minute survey. So we want to know what you thought about today's program. And by the way, that also includes those of you watching this later, watching the recording. We can't achieve our mission at the foundation without your input, without your feedback. So please take a minute before you log off and grab lunch to at least take a picture of this and call up the survey. And while you're doing that, I just want to say that I'd like to thank all the physician researchers, me and on behalf of all my colleagues at the Rare Kidney Disease Foundation, who continue to broaden and deepen our understanding of ADTKD, and who work tirelessly, as you've heard, to find a treatment. And also thank all the clinicians who share these advancements with their patients in the form of earlier diagnosis, effective treatments, and improved patient care. And with that, I'd like to invite you all to return tomorrow at 10 a.m. Eastern Daylight Time for our patient program. You'll hear from additional noted experts on managing symptoms of ADTKD, options for genetic testing, and helpful hints for patients on how to approach the conversation about genetic testing with your nephrologist. So until then, we're about to log off. Take a photo of this, please, and fill out our survey. And have a great afternoon or evening, and we will see you tomorrow. Thank you.